Yesterday, basically, we had a discussion on the brief discussion on the biological sequences, and especially the marvelous presentation made by Professor Devaraju. He's also one of my friend. We had together lot of experience in the under the Bangalore University. He was before in Bangalore University, and I used to go there as evaluator and as examiner, and that way we became a good friend. Till now, I'm having contact with Professor Devaraju. So he made a marvelous presentation yesterday, and I'm trying to just extrapolate that particular presentation. And I'll try to start with the basically the second topic, whatever given to you, that is the different biological sequence and retrieval the sequence from the databases. And yesterday's one part is left out, that is the med minor. I'll try to touch that particular part also. So let me start with my presentation. So biological database and the sequences or information retrieval from the database. So here basically I'm going to talk about brief idea about the biological database, what are the different databases are there and how these databases are developed. And finally, I'll give some kind of practical aspect. I'm having some kind of screenshot, step by step I put it in my this particular PPTs. And you will come to know that if you wanted to get some kind of information by using this particular biological database, what kind of step you're supposed to follow. So biological database, so biology is a data-rich science, all of you know, because a lot of in our genome itself, if we can consider the huge billions number of base pair, it is packed in a small particular region inside the nucleus and inside the chromosome. So if we opening and one by one that particular code, and if we try to get the information from it, we will be having like an ocean kind of information. And every researchers nowadays in the biological field, they are ready to work on the molecular biological aspect. And we are having a lot of machine learning process. So sophisticated computational method also we are having. So by applying all those particular instrumentational process, we can generate huge number of information related to the biological sequence. So biological sequence, it could be of nucleotide sequence, it could be of protein sequence. So huge number of informations we can gather within short period of time. Now, it was a challenge before post-genomic era. So how to reveal the genetic code and how to, what kind of nucleotides are there in the particular gene and how they are encoding the protein. That was particular challenge. And after post-genomic era, so it is one more challenge for us that whatever the informations we are preparing, whatever the biological informations we are getting, because that is coming from the different researchers. So how we can store those particular information, and those if we are storing those information, definitely next time if some group of people are really interested to do in the same kind of work, so they can take help of our previous work and they can follow that particular work, and it will be easy for them to not to repeat that particular same kind of work. So for that we required some kind of database, or we required some kind of software. In that software, now we are storing all sort of biological informations, and these biological informations whenever we required, by putting the specific keywords or specific related works, we can retrieve that informations whenever we feel it will be useful for us. So that way the databases have started creating, and after 1986, 1987, these databases science have started creating, and we are thankful for the computer science people so who have worked along with us, and they have created this particular biology databases. And we are just create whatever the sequence related information nowadays we are creating, that sequence information directly we are providing them, and they are storing with the help of computer algorithm, computer knowledge, they are storing that information into all those particular software. And those software are having huge information. So it is all these softwares are basically public accessible software. So anybody in this world, they can able to access these particular softwares, and they can use this information whatever present in the software for their research purposes. So that was the onset of to creating the biological databases. Actually, biological databases, as I mentioned, after 1986, 1987, that concept has come. But before that, other kind of databases informations, this computer science people have already created. So we have just taken that help from them, and we have expressed our knowledge to them, or we have shared our knowledge with them, and they have understand what we are actually looking for, and based on that they have created that specific algorithm, and with the help of this algorithm we are storing it. So biological database, as I already mentioned, biology is a data-rich science. So biological database, basically it is the computer software containing an organized body. Basically they are keeping the biological informations, and every day basis they are updating this informations, and based on our query they are providing the informations, and we are using that information for our research purposes. So biological data, wherever we are keeping, this is nothing but a particular software, and some people definitely are working on this particular software regular basis. They are updating, and if you are opening the Google, if you are typing some kind of word, or if you are not opening their particular URL or that particular database, that particular software. So if you are providing the specific keyword, with respect to the keywords, there will be having lot of your processing of our data. So what exactly we are looking for, so that from this particular software or the database, they are going to find out, and ultimately they are going to present that particular related information in front of us. So there will be having lot of data mining aspect. So all this data mining, what is the data mining, I'll be telling. So based on this data mining aspect, basically the databases are going to provide us the information. So so huge information every day basis we are keeping. Like every day, if you look at the NCBI, so today how much informations are present, after 1 hour or after 2 hours, we can see it will be at least 10 to 15% increased. So you can understand there is having huge demand to store this information. That's why we are storing in all kind of databases our information. So all these databases are having specific objective. What is the main objective? Objective is to providing that sequence related information to the various kind of researcher. And where today, if I wanted to know that about a particular protein sequence, what kind of structure it is going to form, what kind of functional activity it is going to make. So I can knock the different type of protein database. My sequence I can compare with those particular proteins database sequences, and the protein database sequences already having some information stored. My sequence, if it is matching with those particular sequence, I can predict my sequence also must have such kind of structure and the function. So if you wanted to know the specific gene segment of the genome, I can use the specific database, and I can get the information related to the particular gene, related to the particular gene sequence, and what kind of protein the gene is going to make. All aspects I can able to get if you are using such kind of databases information. So every database is main agenda or main aim to provide the all kind of solved biological sequence related information to the researcher. Now these biological databases are having different classification. So this is as per our own convenience, we have made this kind of classification. So these classifications are telling entire biological databases will be of 3 types. One is the primary database, second one is the secondary database, and third one is the specialized database. So primary database, so here if I'm doing some kind of biological research, I'm having one particular protein, I have sequenced that protein, or I'm having a particular nucleotide sequence. I have after sequencing, I got those particular alphabets. So those alphabet directly by putting my name, my designation, my co-guide or guide, if some researcher associated with me. So I'm putting all the name with respect to that particular sequence, and I am putting those sequence to this particular database. So when I am providing this my sequence with this database will ask me a lot of queries. So once we are able to satisfy those particular queries by able to give the proper answer of those queries, so definitely they are going to store my data into the respective database. Now, if I'm having the protein sequence, I can store it to the protein database. If I'm having nucleotide sequence, I can store it to the nucleotide databases. So again, what kind of biological molecule I'm using, based on that, it is a protein database or nucleotide database, different name we are having. But main aim of this primary database to store the raw data or raw information about your sequence. Best example, we are having GenBank, we are having Protein Data Bank, PDB. So in the GenBank or even NCBI also part of this primary database. So GenBank or the protein database, GenBank, this is related with the nucleotide sequence, and PDB's Protein Data Bank, it is related with the protein sequence and the protein structure. Basically, they are taking care of the protein structure. So whatever the proteins X-ray crystallographically structure or NMR structure we are determining, that information we can store into this PDB. Whereas the GenBank, just to nucleotide sequence related informations, we can able to provide them and they can able to store them. So these are the primary database, because whatever the informations we are having directly, we are putting them, we are storing there. So this is the primary database. And secondary database, it is depending on the primary database information. So for my research, if I wanted to use the primary database information, I can able to get the information from the primary database, and based on my research aim, based on my methodology, whatever I am following, based on that, I am modifying the data. I'm extracting some information with it, and I'm adding also some information with the primary data. And then if I'm keeping it, that will be considered as the secondary data. And the database which are keeping them, these are the secondary database. Like for example, we are having the Swiss Prot, we are having protein information resource database. So all these based on the primary database, they are using the primary database information, and based on the methodology, whatever they followed, they are modifying the data and keeping that. And specialized database, this is also once again very important. And the specialized database basically taking care to store all sort of your genome related information of various type of plant, various type of bacteria, various type of virus, or it could be of insect, any kind of living thing. Okay, their entire genome related information basically we are putting into the specialized database. Like we are having Arabidopsis thaliana genome database, we are having human genome database, we are having microbe like E. coli genome database. So these are the specialized database. So this specialized database, it is a special kind of information they are creating or we are storing there. Like fly base, all kind of fly related information they can keep. HIV sequence database, all sort of variations of HIV strain, whatever is there. So all sort of information they are keeping. Even ribosomal database, all living system, what kind of ribosomal 16S or 18S components are there, all related information they are keeping that particular databases. So as I mentioned before, based on our usefulness, based on our own activity, we have created this kind of group, primary, secondary, and the specialized databases. So these are the URL of all important databases. So if we can click on this URL directly, this particular database we can able to reach. So these are the sequence of biomolecule database like DNA, RNA, the protein related. We are having GenBank, EMBL, DDBJ, Swiss Prot, PER, etc. So these are the related web link. Then we are having biomolecular structural database, PDB, Protein Data Bank. So directly we can click here, we can reach to the PDB. Then bibliographical or scientific literature mediated database. So yesterday, Devaraju sir has explained very nicely PubMed, how to use it. So apart from PubMed, we are having the Scopus, we are having some other kind of literature databases also. But PubMed is very much connected with the NCBI. So I have put it here. So we can access the PubMed by clicking the NCBI, and through the NCBI directly you can reach the different type of literature. Then we are having patent database. So patent database also kind of your special kind of database where all sort of patenting informations we can able to see over there. And this patent database, basically whatever I have put it here, this is related ASPITO. It is called ASPITO. Okay, so this is the database basically keeping the information with related to the different type of expression vector, different type of your plasmid, artificially constructed plasmid or artificially created some kind of gene compositions. So all related information, this patent database they are keeping. And then metabolic pathway databases, that are KEGG, Kyoto Encyclopedia of Gene and Genome. So here, if we look, we can get all sort of information of any biological metabolic process, what kind of enzyme and gene involved, what kind of enzymatic accession number, those particular part they are having, etc. So all sort of information, basically we can get it from the KEGG. Next, we are having gene expression profile, EFP browser. Here, basically these are useful for our microarray database. If you wanted to know that this one particular gene with respect to a particular conditions, how it is expressing, we can able to get that idea. Some noise is coming because some construction work is going on outside. So genetic disorder, like we are having OMIM, all sort of onset of Mendelian inheritance. So all sort of Mendelian gene related inheritance related information we can get it from the OMIM. So this is also kind of special kind of database. Then whole genome sequences databases we are having, like Entridge and genomes itself. And Entridge, once again, it is connected with the NCBI. So when we can access the NCBI, there is having alternative method. We can knock the door of Entridge, and through the Entridge also we can able to access the information from NCBI. Next, we are having educational database like teaching tools, many plant cell, how it looks, bacterial cell, how it looks, and animal cell, what it looks, and different animation also you can found in this particular educational databases. So as I mentioned, the biological databases, the concept has came a bit later, but before that, computer science people, they have already created various type of databases, and we have tried to give our knowledge, we have shared our knowledge with them, and ultimately they have created this kind of biological databases for us. So primary databases, as I already mentioned, this is keeping the information related to the protein and the nucleotide sequence of our research, daily basis research, whatever you are doing. So from there, if you are getting some nucleotide sequence, protein sequence, so we can able to get those particular data. We can able to keep those particular data in these primary databases. So this is the DDBJ, DNA database of Japan. This is only one particular database so far we are having, and this is the Asian continent. This is the only one particular database. So from Japan, they are taking care of different type of nucleotide sequence, and they're storing there. And of course, that is connected with the EMBL, European Molecular Biology Lab. So you need to remember, so whatever the databases information I have provided here, so all are connected with each other. Okay, so what are the special databases are there? Some part also is connected with the primary database, and primary database information, again, it is connected with the secondary database. So all are having one interlinking connections, right? So whenever the any information that suppose I am looking for OMIM, if I do not have such information, might be somewhere that information kept to the your PDB, or it is kept to the DDBJ. So immediately it will extract that information from DDBJ or the Swiss prot or the P, and it will provide to you. So that's why I'm telling these are all interconnected. So whatever the information we required, we can get the information from any one of these databases. And so primary database like GenBank, EMBL, DDBJ already I mentioned. PDB, that is protein, already mentioned, X-ray crystallography and NMR mediated. Whatever the protein structure we are solving, that particular structure we are putting to the PDB. And secondary database, it is of course depending on the primary database informations. So you just look at, I'm just giving example like Swiss prot. If you are using the Swiss prot is keeping information related to the particular nucleotide sequence, and with this particular nucleotide sequence, what kind of protein it is going to make, and the protein could be what kind of sequence and what kind of function that particular protein is going to make into the biological system. So all sort of information SwissProt is keeping. Okay, even Trimble also keeping the same kind of information. So what is the difference we found here with the primary database to the secondary database? In the primary database, only the sequence related information we can able to store. Whereas secondary database, if we open and if we here with the respect to the primary database, what sequences are there, that sequence, what kind of protein it is going to make, protein, what kind of amino acid will be there, and protein, what kind of structural component it is going to make, and which particular part of that protein will be the active site. So all sort of informations will be there in the SwissProt. So this is completely depending on the primary databases information. And the Trimble, this is also similar way, they are keeping the information, but Trimble only it is useful for nucleotide informations. They are with that particular nucleotide or the gene, if it is nucleotide is coming from particular gene, so the gene, the DNA, what kind of structure will be there, and what kind, if it is RNA, what kind of folding should be there in that RNA part. So that information only they will be storing into the Trimble. So you can understand that secondary databases, what is the function, and specialized databases like flybase, wormbase, ACDB, TATE, these are the different gene name with the gene, similar gene if we found in the all kind of dicotyledon or monocotyledon or all sort of plants. So what kind of variations will be there among them, composition wise, everything to be stored there. And worm based, all kind of worms, okay, that related informations will be there. Fly based, all kind of fly, that is insect related informations will be there. Even some microarray databases are there. GOE also it is there, Gene Omnibus. So these are all also part of your specialized database. So whenever we require to tally our microarray result with the databases available microarray result, so we can use this kind of microarray database. Otherwise we can use the GenBank EST database, express sequence tag mediated database. So these are all specialized kind of database. Recently we have developed it. And a gene expression database that is already mentioned, this is connected with the EBI, European Bioinformatics Institute. They are basically taking care of it. And these are the few example with the your different hyperlink like SGN, that is the Sol Genomic Network. So it is keeping care for tomato, potato, plant, pepper, petunia, all sort of plant related information we can get by knocking the door of this particular database. Legume based databases we are having, so all sort of legume plant related information they are keeping. Bean genes, that is all kind of Fasciola vulgaris, all kind of beans, that particular family plants information we can store from here. Graminy, it is specially keeping information all cultivated crop, including the rice, maize, barley, sorghum, pearl, etc., oats. Then tiger plant transcript assembling database. So different type of plant species is transcriptional level information. That means what kind of mRNA it is encoding, mRNA, what kind of sequences are having, that mRNA how much long, all information they are keeping here in this tiger plant transcript assembly databases here. Aphid database, so multiple aphid species information. Cystomonas, this is information about the biotechnology of the Pseudomonas. So this is actually created by looking the activity of the professor, late Professor Anandamohan Chakraborty, so who has created the super bug. So later on, the people have started working on the other group of Pseudomonas, and all sort of information you can get from the Cystomonas. Then human aging genome resources. So HAJAR, we can consider HAJAR. Okay, the pronunciation will be HAJAR, not GR, HAJAR. So biology and genetical aging of humans with respect to that, what kind of consequences may come, medical complications, what may come with respect to the aging, such kind of informations you can get it from here. This is also medical informatic database. And fly-mine like Drosophila, Anopheles genome. So all sort of information you can get. So Drosophila is considered as the queen of genetics. So it's related informations, which gene, how it is expressing in Anopheles mosquito, what type of gene is expressing, which stage it is expressing. So all kind of informations we can able to extract from this kind of specialized databases. So what so far, whatever the things I have discussed, so we have discussed about the biological databases. So the databases, some they are keeping the information of the only the structure, some of the keeping information only the sequence, and some of them keeping information about the structure function with everything. So we can summarize by biological database this way: some of the structural database, sequence related database, and other database. Structural database like PDB, SCOP, CATCH, and sequence related database like protein, nucleic acid. Again, two different type of sequences biomolecules they are having. Protein sequence related database like SwissProt, PED, Tremble, ProCite, PEFAM. Nucleic acid like GenBank, DDBJ, EMBL. These are the three major one. And other database like Fly database, microarray database, whatever I mentioned, these are the few examples. So this is the summarized form of the biological database. So we can extract the information from all these kind of databases, fine. And as I already mentioned, these databases are interconnected. So protein databases, so if you are knocking the PED, so PED also will be connected with all other kind of protein databases. Like nucleic acid databases, if you are opening the DDBJ, if that information is not stored in the DDBJ, so it will try to extrapolate the information from GenBank as well as the EMBL. Fine. So that way, all the databases are interconnected, and by this interconnection mode, making our research easier, we can get a lot of informations whenever looking for doing any kind of molecular biological activity. Now I'll talk about the data processing. Actually, database, how we can knock the door of the database to collecting the information. So for that, either we need to open click the URL, and after clicking the URL, you need to some places will come that is called the dialog box. You can put the information, or you can need to type there what you actually looking for, and the database will try to processing that information, and it will be presenting in front of you. Suppose you are a layman, you do not know how to use this database. So just in the Google, if you are typing one particular word, definitely from the Google also they will be connecting with the respective databases, and from the database also they will be gathering the information, try to give you. But by just typing in the Google and getting the information is not the authenticated one, and maximum time you will be getting very wrong information. Wrong information in terms what you have typed, it is going to give some other kind of information, but those information is related. Fine. So data processing, when we are opening the particular open-based server of this particular databases, and if you are typing the specific word, what kind of activity is basically going on in the background, and after doing that, how that particular databases are presenting that particular data related information. That I am talking in terms of data processing. So generally, this data processing is a part of the data mining. So data mining, yesterday somebody was asking the question, what is the relations with the data mining with this particular databases? So without data mining, databases accession is not possible. So if you wanted to access the databases will be working based on the data mining, and the data mining concept will be working based on your keywords. What type of words you are typing by using your keyboards. Fine. Suppose I wanted to get the information about the human genome chromosome number 16. So you need to type properly the human genome and chromosome number 16. So until unless you are typing it proper way, software will be getting some kind of confusion like information. So whatever the information is there related to the gene 16 and human genome, everything will be dumping in front of you. So you'll be confused. So which is actual and how to get the actual information. So for that, it is very, very much necessary to type the proper keyword, and based on the keyword, the data mining activity will be continue, and this particular informations will be getting in front of your monitor. So when we are typing a specific keywords, so this is the activity it is going on background of your monitor. That means the software or the databases, whatever is there, they are following this particular process. So user specified data, that means whatever the keywords we are typing, that is the user specified data. We are just providing the data, and data will be there will be the three layer. So one is called information layer, second one is the analysis layer, third is the modeling layer. In the information layer, again, so database is information, pathway knowledge, experimental data integration. So what kind of word I have used, and databases, what kind of information they have stored. So they will try to correlate with your keyword with their first information layer. So databases information, first which databases that information kept. Pathway knowledge, whether it is your metabolic pathway related some information you want, or you want only particular protein related informations, or you want any kind of mutation related informations, what kind of informations you are looking for. So that is called the pathway knowledge. And then experimental data integration. So who have done this experiment, what kind of experimental data they have stored in the primary database, in the secondary database, any other modification on that primary databases information have been done or not. So all will be taken care in the information layer. And then once the information layer is passing, it will go to the analysis layer. So suppose I give the example, human chromosome number 16. Here 16 chromosome number related information, or many experiment, many people have already done. They have kept the information in the various databases. So all information first in the information layer, the software will be collecting. And then the next layer will be the analysis layer. Here they will be analyzing actually which is the authenticated one. They'll be trying to retain those one and try to remove that other related but unrelevant with your particular keywords. So they'll be making a particular table like information, or they'll be providing the information like number 1, number 2, number 3, number 4. So the first information is very reliable and very proper, accurate information. Then second one, third one, fourth one. If you go down, the information will be it will be related, but it will be not actually connected with your keyword. So that analysis layer will be doing that activity. So here will be network analysis, statistical analysis, what kind of statistical activity software is doing. So based on that, it is telling this is the 2021, this particular literature published. So I need to put the 2021 first, then 2020, 2019, like that. So that will be the statistical analysis layer. And then it will be the modeling layer. So modeling layer, whether your data will be presented in the graphical form, whether your data will be presented just tabular form, whether your data will be presented in the pie chart or bar diagram, what kind of presentations you are looking for. So that is going to happen into the modeling layer. And finally, they're creating the hypothesis and simulation. So hypothesis and simulation, everything if it is done before also presenting that information in front of you. So there will be the hypothesis checking, because all software, when you are writing the algorithm, there will be particular hypothesis. And based on the hypothesis, we can use the computer language. We can provide the instruction to the software, and based on that, software will start doing that activity. So the software, that particular software, what kind of hypothesis is having. So hypothesis, it will be telling, and if everything is matching, then only the information will be coming in front of you. Otherwise, they'll be telling, sorry, unrelated, you change your keywords, or you this kind of data is not available, or you have typed the wrong information or wrong things. So that kind of things will be coming in front of you. So you can understand the data mining, how it is important to retrieving the information from the databases. So I'm repeating it again. So it is completely depending on your keywords, what kind of keywords you are giving, what kind of Boolean form, that is a Boolean algebraic form, whatever you are giving there. So based on that, actually you are going to get the proper, relevant, accurate information from these databases. So this is called the data processing. Data will be processed in proper way, and then it will be going to present in front of your monitor, and you can use those data for your research purposes. So as I mentioned, these are all interconnected. I mentioned here, these are all interconnected. So look at here, this interconnection. So the OMIM, if you are knocking the door of OMIM, it will be connected with the PubMed and several other electronics journal, nucleotide sequence databases, even 3D protein structural databases, taxonomical databases. So with all, okay, these are indicating. So any one of these databases, if does not have information, so it will knock to everybody. It will try to extract the information, and here this particular analysis layer, information layer, modeling layer will be working, and finally that information is going to present in front of you. So I hope with this you got the idea. This is what are the different biological databases we are having, and what is the role of these databases, and what is the meaning of data mining and data processing, how it will be happening in the particular software and the open base, or if it is purchasable, so it will be following the similar kind of your protocol or procedure. Now I'm coming to the specific example. So how we can retrieve the information from all these kind of databases. So suppose you wanted to search one particular literature, one particular journal which related to the nucleic acid. Fine, NAR, nucleic acid research journal you are looking for, because your research is having certain aim to get some kind of paper or literature from this particular your journal, and you wanted to use those particular information for your research purposes. So how and what could be your first logic and what systematic process you are going to follow here? So this question I am putting: categorize the databases from the database issue of the nucleic acid research journal based on the current academic year, like primary, secondary, composite, and integrated databases. So here, once you are opening this journal, and what kind of papers you are getting from here. So those paper, once you are going through, how you will able to make this kind of idea, this is my primary information data, this paper is giving my secondary information data, this paper is giving my composite or integrated information data. Fine. So whenever any paper is providing in this particular your any kind of journal, so the latest paper and most important paper, if it is electronic journal, they'll be putting first, and then secondary, tertiary, like that it will be continued. Fine. So suppose if you are using the NAR, nucleic acid research journal, so this would be the your hyperlink for it. So Oxford University Press it is publishing. So if you are clicking on it, and if you are opening this particular nucleic acid research journal, so you are going to get, this is the first home page about nucleic acid research. So this is the journal cover page. So this notes or this particular slide I prepared quite a long year back. So that's why that time, whatever the information I got, I have pasted it here by taking the screenshot. And it was 2013 when I made this particular example. So you can see here 2013 database issue only, it is mentioning here. And if we today 2021, so if you are opening this particular nucleic acid research journal, you can see here 2021, and then below all respective years will be coming. So anyhow, this is the nucleic acid research journal. So this journal about the instruction of the author who is going to publish their paper. So they need to follow some kind of instruction, even editorial, how much they need to pay it, and what is the procedure, how they need to provide the reference, etc. will be there. So I'm not talking about that. I'm just talking how to get the information from here. So if you're opening this particular journal, right hand side, you can see a lot of web related issues are presented. The web related issues are there. So year wise, many informations they are providing. Fine. So now here you can see that 2013, suppose this is 2013. So 2013, whatever the information is given, that will be my current information. So you can click on the 2013 database issues. So all sort of paper, whatever going to get, those will be your actual your authenticated, means with respect to your research. I mentioned that will be integrated or the composite informations you are getting from those particular paper. If we are going to the exactly after the 2013, if we coming to the 2012, you are whatever the paper information you are getting, you need to consider those are the secondary information paper. And whatever the means 2010, 2009, if you are going back, so whatever the paper related information you are going to get, you need to consider those are the primary databases, primary information related to your research. So this way you need to search one particular important any kind of journal, and you got the idea what is the meaning of primary, secondary, and composite database, and you need to extract the information. You can create the folder accordingly that, and you can upload your abstract as well as you can download it paper in the respective journal, and you can sort out your research related activity in that way. So this above page, whatever you got, it displayed on which here is a hyperlink 2013 databases issues are there. And here all it is connected with the NCBI, DDBJ, EMBL with the everyone. So if you are clicking on the 2013, so these are the articles you are going to get. Right? These are the articles you are getting. So these articles are Of course, some of the full text will be available, some of these full text will not be available. Just a minute, very much noise is coming. I just tell them because outside not here. Just a minute. Sorry for these interruptions. Am I audible? Hello.
Yes, sir. Yes, sir. Audible.
Fine, fine. So database is issued 2013. If you are clicking, you are going to get these are the various article. So some of the article will be full text, some will be in the PDF form, some will be in the HTML form, and some will be not be available into the full form. Abstracts will be there. So when you are taking the information from the abstract, you need to remember you need to take the entire author name, the journal name, which year it has published, everything. Okay, it is not like that only the abstract, whatever the words has mentioned, you are copying and you are keeping for your future purpose. No, that is not the way. You need to take the entire information about the articles, author, year of publication, with if it is DOI number, if it is there, with each and everything. And sometimes though it will be 2012 or 2010 related research paper, if you are going through that particular research paper, you may found it is having direct relations with your aim of the research. So in that case, you can take that particular paper, you can put it to the latest composite folder, whatever you have prepared in that. So this is the simple way to search any kind of your journal, which could be nucleic acid research related or any kind of journal. So you need to look at the right hand side, this particular year wise issues, and you can get the information from this journal this way. Next, I'm coming to the nucleic acid sequence retrieval. So this is actually related with your bioinformatical activity. So nucleic acid sequence retrieval, you can use the GenBank, you can use the NCBI. So we used to tell NCBI is the brain for like humans, we are having brain. Without the brain, we cannot able to do any kind of activity. So NCBI is a kind of brain for bioinformatical activity, or the bioinformatics subject we used to tell this is the brain. So NCBI, that is the National Center of Biotechnological Information. Here you can you can connect with all kind of databases, and you can get the information for all the nucleic acid, protein, structural, functional aspect, your microarray data, all kind of activities connected with the NCBI. And this is the URL www.ncbi.nlm.nih.gov. NCBI, National Center of Biotechnological Information, National Library of Medicine, National Institute of Health, and this is the government institute of USA is taking care of it. So this is the authenticated 100%. Yesterday, Devraju sir was telling .gov, always you need to prefer. So you can use this hyperlink, or directly you can type on the Google NCBI, and whatever you'll be getting, just click on that, and it will be taking to you to the NCBI homepage. So this is the NCBI homepage. So here in the NCBI, here it is written all database. So if you are clicking over here, you will be getting here n number of list items here. And if you look at the left and the right hand side, so NCBI homepage, so I mentioned already DNA database, RNA database, even protein, microarray, all sort of information. So we can also able to extract from the NCBI. And right hand side, different popular resources, PubMed. If you wanted to study the book, different bookshelves are there, PubMed Central, PubMed Health, Blast, etc., many informations are there. So when you are going to extract the information from NCBI, you need to use, this is called the dialog box. In the dialog box, correct way, you need to type the particular keywords. And if the keyword is related to the nucleotide, you need to click over here. You can, in whatever the list item will be there, in that nucleotide will be there. You need to select the nucleotide. So nucleotide will appear here. You write the information about the gene or sequence, whatever you are looking for. And then you can click on the search. So if you are, suppose I'm typing like nucleotide I have taken here, actin and aphid. I have typed here and actin and aphid. So, and this is the Boolean form or Boolean algebraic form. So I'm putting it here. So as already mentioned, So how I am putting the accurate keywords, based on that I'm going to extract the information. So I'm putting it here, and I'm clicking on the search. Once I'm going to click on the search, you need to remember this NCBI is the primary database. So here all sort of informations will be there. So you have typed here actin and aphid. So whatever the actin related information, whatever aphid related informations, as it is the primary database, everything will be coming in front of you, right? So until unless you are providing the proper Boolean operators form, you are not going to get the actual information. So if suppose I am writing only actin comma aphid, that also is not going to give the proper information. And until unless we are putting the and, that is called the Boolean operators form. Until unless I am putting it here, I am not going to get the accurate information, because I am repeating it, because NCBI is the primary database. Here all sort of relevant information which is exactly will be matching with your aim of work or not, all sort of informations you are going to get from there. So anyhow, so actin and aphid, if you type it properly, so in front of you, this kind of result page will appear. So this is the number 1, number 2, number 3, like that. This page could be sometimes you can see here 110. These pages are there. So this is indicating, as I mentioned, this is the primary database. So based on the aphid and all those, so if latest information will be coming first, then second latest, third latest, like that it will be presenting in front of you. And all this will be hyperlinked conditions. Fine. So if you click on this hyperlinks, it will be taking to you that particular information page. Otherwise, you can see here it is written with respect to this particular genome or whatever it may be. So it is how many nucleotide base pair it is present, and then it is given the accession number related to this particular sequence. And here it is having overall view, then FASTA format. And then if you are looking for a graphical format information about the sequence, everything we can get it here. So either you need to click on the hyperlink, or separately if you click on this particular gene related information, it will be taking to the same page. Otherwise, if you click on the FASTA, the sequence, one special kind of format, it will be appearing in front of you. And for all kind of biological analysis, we require this FASTA format. And the graphical format also, if you click on that, a graphical view will be coming in front of you. And so this way the result will be appearing in front of you. And actually from here only your actual research work will start. Fine. So you have opened the NCBI, you have whatever the aim you are having with your research. According to that, you are typing the word, and what are the relevant information is coming. Now you need to find out, and you need to go through those information, and you'll be getting the idea which one is useful for you, which one is not useful for you. So every sequence, when I mentioned in beginning, so when I'm getting some protein sequence and nucleotide sequence from my research work, I'm submitting them to the NCBI. I'm submitting to the GenBank or any other kind of sequence databases. So once I'm submitting it, so immediately they are going to provide some kind of submission number and some queries they will be having. So I need to satisfy it, and that sequence once again they'll be checking. And after checking, if they found it is perfect, they'll be mailing you by providing the accession number. That accession number, this is the accession number. So this accession number or the GI number, that is the GenBank accession number, and this accession number is the whole NCBI accession number. So this accession number is telling you this is your created informations. So if you know this particular accession number, here if you putting this accession number directly, it will be taking you to this particular page. Whatever the information is given here, it will be taking you that particular page. So it is somehow like a library if you are going. So every library, every book will be having certain kind of catalog and accession number. If we able to tell the catalog number or accession number to the librarian, so they will be searching you and they'll be giving to you. And if you do not know what is the accession number, and if you're looking for any particular molecular biology book, so you need to go to the molecular biology rack and need to hunt it, which is the best for you and which is actually having the information related to your activity. So it is like that. If you are having the accession number, directly it will be giving to that particular book. And if you do not know accession number, just type in this way, all related sort of informations you are going to get. Now, if you are clicking on any of this hyperlink, immediately this kind of result page will appear in front of you. So this is the informations related to your sequence. And if it is a nucleotide sequence, definitely you are going to get the information like what kind of protein it is going to encode, what is the particular exon introns part, where exactly the protein coding sequence is starting, and what could be the protein sequence. At the same time, what is the nucleotide sequence you have submitted, all sort of informations you are getting here. So this is the reference number or accession number. You can see this is the locus, which particular gene. This gene is having total 1773 number of base pair. Then this is the again accession number. This also you can able to use which versions of the software and gene bank they have used. They have mentioned here, and this is called the primary informations about your sequence. And then the organisms. When we are submitting the sequence, suppose I have isolated the sequence from Pseudomonas particular strain. So I need to provide the name, the Pseudomonas so and so strain. I have isolated the sequence I'm providing you. So that sequence informations, that organism information you need to provide. And then this is the author name who all have worked together. Even sometimes from which environment I have isolated it, which particular protocol I have followed, all those information I need to provide. And then that comment, this is their comment. Comment means whether I have published it already, or I'm going to publish, or this is very newly invented one particular species. All such information along with what kind of sequence, like a pyro sequencing I have used, or Sanger method I have used, or some dideoxy method I have used, what kind of method I have used, all sort of information I need to provide here. And then we'll be coming to the primary information. So primary information you look at here. So first 1 to 241 up to this much one protein coding part is there in my provided sequence. Then 240 to 200. 1372, then 1373 to 1759. So these are the sequence. Actually, these are the part, because I mentioned already that how I am writing the particular algorithm to start off this particular software. Based on that, it will work. So the software is also having certain kind of limitations, how much sequence they are alphabet they are considering at a time, to analyzing and to finding whether specific exon and introns, or if it is the prokaryotic sequence, there definitely only single ORF mediated encoding protein will be there. In that aspect, exon introns concept will not come. So that's all of we know. So I'll tell you that when we predicting the gene sequence, so how software these algorithms are based on that algorithm, how software is running. So this is the information they are providing. And source, this we can look at here. So nucleotide 1 to 1773, this is total is there. And the gene, okay, gene informations I am trying to find out from entire 1773 nucleotide, whatever is there with respect to that. So here the CDS, this is important. CDS is indicating this is actual protein coding part in this particular provided nucleotide. And the CDS is indicating, so this is the protein coding sequence. Okay, this is the protein is going to encode from this particular sequence, whatever I have provided. So this you can see here, this is the protein amino acid. So amino acid with the capital alphabet, we can represent the all 20 amino acid. So these are the amino acid, how one after another it is going to encode from 2033, from 242 to 1372. So this is actual 242 positions. It will be your starting codon, and 1372 position, it will be your stopping codon, whatever the nucleotide sequence you have provided. And based on that, they found this is the actual protein it is going to encode. So this information also you am getting. And below, this is the NCBI actually GenBank format. So in the GenBank format, this nucleotide will be represented with the small alphabet, ATGC. It is all a small alphabet, because protein they are going to represent with the capital alphabet. So there should not be any confusion. So it will be like that. And every line, if you look, it will be having 60 alphabet. 1, 2, 3, 4, 5, 6. So 6 column or 6 Group is having 10 alphabet, so total 60 alphabet will be there in one line. And next line is 61 to plus 60, then 121 plus 60. So ultimately like that, it will be reaching to your completely whatever the last nucleotide you have provided, it will be keeping last nucleotide, and finally there will be having double dash. So this is the informations I can extract from this particular, any one of this particular hyperlinked gene. Okay, and all are of course actin and aphid related. So I can click anything here, and once I'm clicking on this hyperlink, I am clicking on the gene bank. Ultimately, it is bringing us, taking us to this particular home page where you need to understand what is the meaning of it. You need to get the idea what will be the starting codon, stopping codon, what kind of protein it is encoding, and this alphabet, why they are exhibited or presenting in the small alphabetical word. So this is the NCBI, and from the NCBI we can get the information. And if you want, I mentioned the FASTA format is very important for us to analyzing all kind of software, means all kind of bioinformatical activity. So you can click over here FASTA, it will be taking you to the FASTA format of the gene sequence. Otherwise, you can click over here this FASTA, it will be taking you to the that particular FASTA information related gene sequence. So you need to copy that gene sequence, and you can use for knowing the structure or the what kind of protein it is going to encode, and the protein what kind of secondary structure, completely structure will be there, all kind of activity you can able to do with that. Next, I'm coming to the bibliographical database. So this is already nicely Professor Devaraju has explained yesterday, so I'm not going to touch this topic, bibliographic database. So with respect to the NCBI, that PubMed is connected, PubMed and PubMed Central, and from there only we can access this particular bibliographical or literature related databases. So I'm just skipping this particular part. I'm coming to the next part. There is a whole genome sequences databases. So if you wanted to get the information about a particular organism's whole genome, so that time you need to knock the special kind of databases. So that is a whole genome sequence databases, and that also you can able to access through the NCBI. So that's why I mentioned NCBI is the brain for our bioinformatical activities. So you need to open the NCBI. You can once you're opening the NCBI, you can see here genome and map. I've highlighted here. Here is having genomes. So directly you can click on the genomes. It will be taking to their particular home page, or else you can click on the genomes and the map. So first I'm talking on the genome. So suppose you are clicking on the genome. So once you are clicking on the genome, it will be taking you to this particular home page. And here, suppose I want to know about the microbial genome. So here the genomes is already mentioned. You need to type here microbes. Otherwise, you just click over here the microbes. If you wanted to know the genome of Pseudomonas, genome of E. coli, so that time you need to type here E. coli and the search. So it will be giving you the E. coli genome related informations, or else you just here the microbes option will be there. So you just click on the microbes. Once you are clicking on the microbes, it will be taking you to the microbial genome page. In the microbial genome page, in the right hand side, it will be written very clearly so far how many microbe genome has sequenced completely, and what kind of, how many genes are present there, what kind of protein it is going to encode, all sort of informations we can able to get it here. Like that, if we type the human genome, so it will take you to the human genome page, how many chromosome, which chromosome, what gene, total base pair, every sort of informations you are going to get. So this is the example I had given with respect to the microbial genome. And if you clicking on this particular genome and maps, this particular hyperlink, so immediately it will be taking you to this particular page. Don't look at right now this one. So it will be taking you to this browse genome page. So you need to click here organism bacteria. If you want, you can click the bacteria. And if you require the completeness, that means all bacteria, whatever is there with all of them, you wanted to get some kind of phylogenetical tree related and sequence related information. So you can leave it like all, and collapse level like a limit by class. That means so whatever the suppose 1,000 different bacterial informations are there, you want to get only the 10 or 15 or 20. So that time you need to mention that particular number. So after that, if you are clicking it, here will be their enter button. If you are clicking it, So whatever the number of bacteria mentioned, so it related to that bacteria, what is the relations, which bacteria, suppose E. coli 5 member have sequence and they have deposited. So 5 E. coli strain related information will be in the one group. Then suppose 10 different Pseudomonas species are there, and different group of people, they have deposited that entire genome part. So that related informations are there. So here, this is not the phylogenetical tree. Remember, this is not the phylogenetical tree. This is just telling you the what are the bacterial sequences are there, which sequences are having how many number of nucleotide, and if they are same sequence, they will be presenting in the one particular group. So this way, if you, this green part, whatever is presented here, if you click on this particular green part, immediately it will be taking you that particular sequence part. Okay, so wherever, whatever the green part is mentioned here, just you can click over it, and it will be specifically hyperlinked. So if you're clicking on it, it will be taking you to the respective gene sequence or the genome informations, whatever it is stored there, it will be taking you directly to there. So this way we can analyze the particular, we can get the whole genome sequence database, and we can access the whole genome sequence database, you can get the information. Next, I'm coming to the organism specific database. So organism specific database, suppose I have put it a questions, what are the gene model known for the Arabidopsis genome? So Arabidopsis related, what kind of gene informations are there? Among that, I want to get the information about the GLA-BRA2 on that. So you need to open, this is a particular Arabidopsis genome related server, open based server. You need to click on that, you need to open it. So once you are opening it, this kind of homepage you are going to get. So here this red part is indicating highlighted part. This is the dialog box. You need to type here what exactly you are looking for, because this is actually Arabidopsis genome related informations only going to provide. So I mentioned put this word GLA-BRA2. So you need to write here GLA-BRA2. This is nothing but a particular gene, and immediately click on this particular search button. So immediately if you're clicking on the search button, so that particular gene related informations, it is going to appear in front of you. And which gene, this particular gene, whoever have done it, that work, and what kind of functions are having. So what kind of keywords will be mentioned here, keyword respected, suppose it is encoding one enzyme. So enzyme, it is ATP dependent enzyme, or it is somehow some related to some metabolic pathway. So all sort of informations you are going to get it here. So you need to look at here that this particular number, okay, gene model number. So I already mentioned here, my question is the gene model, whatever that they are present with respect to the Arabidopsis, this particular gene, I wanted to find out. Because this gene model is very important to my microarray data set analysis. At the same time, this gene model is very important to predict that whether this particular gene part I can able to fuse with the some other gene part, and I can able to express recombinantly that particular protein into the Arabidopsis or on some other kind of plant species or not. So for that, this particular accession number or this particular number is a model number is important. So this model number is indicating what experimental process they have used, and based on that they have analyzed, this is the function of this particular gene. So this model number you need to remember. So that's why I've highlighted here. So suppose I got this particular model information which related to that particular Arabidopsis gene. So I need to note it down this particular model number. And if we click any one of this model, so I am going to get, this is the gene structure. Basically, this is the physical structure of that particular gene. It is going to provide, this is the bar it is indicating. These are the specific protein coding part among this particular gene, and rest whatever the single line it is presenting, that there is having either doubt, otherwise it is not going to encode any kind of functional protein from this particular gene model. Okay, so this way this particular information is going to come in front of you. Now you need to look all this particular three, whatever you got it. So you need to click all these three. You need to look how better way this model have resolved. Okay, if somebody has provided that a complete blue bar, that means their particular group or their particular researcher able to find out the entire gene sequence of that particular Arabidopsis gene. So they able to know, or they able to extract the entire information. So by looking this particular blue bar, you need to consider which particular model you're supposed to accept and which particular sequence you're supposed to use for your work purposes. So this way gene model we can use. And even if you click on this particular bar, okay, this blue bar, it will be hyperlinked. If you click on this bar, again, it will be taking you in this kind of page where what kind of information, so that means what kind of experimental model they have used, that experimental model related informations, and whether they have used any kind of biasness or unbiasedness, what kind of statistical method also they have followed, everything it will be providing in front of you. So you need to go through all those things in details, you need to note it down everything, and based on that you can create your expression vector or any kind of vector for your transgenic plant developmental purposes. And here some external information will be, one column will be there. If you click on the external link, so you are going to get this kind of home page. So what is the function of this external link? You will come to know to the next experiment, that is the gene expression database. Actually, this is going to give you idea about the gene expression. So how better way the gene is going to express, whether in the cold conditions, because I'm talking about the plant. So if it is cold stress, it is there, how better way the gene is going to express. If it is the temperature stress, how it is going to express. If it is the drought conditions, how the gene is going to express. If the humid condition is there, how it is going to express. So that is very important to understand about the gene ontology, about the gene specificity, about the gene's working principle. So you are going to get the information related to this particular model gene, whatever I mentioned, that is the GLABRA2. So it related information, the expression informations you'll be getting here. So I'm talking about the gene expression database. So you are going to do the same step, and you are reaching to this particular part, gene expression database. So once we are reaching this particular part, here I have hyperlink EFP browser and the gene predictor expression data, gene expression prediction data. So these two, any one of it, you can click. These are all hyperlinked one. So if you are clicking on it, so different, this kind of page will appear in front of you. Remember, this color, whatever is indicating, the color is having some significance. So this color is indicating which particular stage that particular gene is going to express and how much the gene is going to express everything. Okay, so suppose the gene is going to express in the sepal, petal, or it is going to express in the stem part, or how much when the stem will be in the green color, that time it is going to express, or if it is a leaf, or it is the seed, in the seeded, if it is seed, whether inner seed code or outer seed code, where it is going to express, how much going to express, all sort of informations, it is going to denoted with this particular color. So this is the if you are clicking on it, if you are clicking on in this EF browser or the gene expression data, so you are going to get this kind of home page. And now here you wanted to look for the seed mediated expression or leaf mediated expression, that you need to mention here, and you can provide the accession number of the gene. If you know, directly you can provide there. Otherwise, Arabidopsis, just you can provide the seed related information and click on the go. So in the database, Arabidopsis seed related information or expression data, whatever is there, it will be presenting in front of you. And you can see this is the color annotation and the color scale. And this is the number. It will be going to tell you that how better way and which particular stage this gene is going to express. And if you wanted to know the graphical pattern of the gene expression, so that option is there. So graphical pattern of gene expression, here will be there. You need to observe that is the in the bar chart or not. That means a graphical gene expression pattern if you wanted to look or not. So if yes, you can click over here. Ultimately, it will be they're giving you this kind of picture. So this color, already I mentioned, this color is having certain meaning. So this particular gene locus, how better way, which particular condition it is going to express. So that is also going to tell. And this is the statistical data. You can see this is the bar diagram, and this is the standard deviation is mentioned. So that means the software, how it is presenting that information is very authenticated, because statistically this is signified data. So you can believe on this, and you can, based on that, you can continue your further research work. So this is the way how you can get the genome or gene related information, expression related information from any particular type of database. And here I just till now I have mentioned that nucleotide, how we are getting the information, how particular genome, complete genome related informations you are getting, particular gene expression information how you are getting. Now I am coming to the protein database, how you are going to get the protein, particular protein related informations. Okay, so XPassay, that is earlier very famous one particular open based source, and now also we can use the XPassay. But if you're using the XPassay somewhere, 2 or 3 different times you need to click on, and then only we can get the directly to the sequence level. And now what we are doing, directly from the NCBI itself, we are extracting the information, or from PDB also one particular protein solved structure, whatever is there, from there also you can collect the sequence information. Otherwise, some other databases also are available now with related to the protein information. We can use that. But here I'm projecting about the XPassay. But because first time when the people are learning the bioinformatics, they must know what is the XPassay and how to handle the XPassay. So protein database, here in this particular exercise, I'm going to explain that how this FAD2 gene of particular oils, this is the oil seed plant, this particular gene is going to express. So what is the sequence of this particular gene? And if we take the sequence from any particular oil seed plant, this particular gene sequence, next stage of it will be, I need to compare this particular protein sequence or this particular gene sequence with the other plant. That will be our next discussion. That once we are retrieving the information, nucleotide information, protein informations, what next we can do? We need to compare it. We can get the after comparing, we can get this kind of inference that these are very closely, these are the homologous, these are the similar percentage wise, this is the similarity gene, etc., etc. Fine. So first, before going to that particular aspect, you need to get the protein related informations. And the protein, as all of you know, it is coming from the gene itself, right? So the gene only will be having nucleotide composition. The gene can produce the mRNA. From mRNA to it can give the protein. So here I'm talking about the FAD2 gene. This is present in the oilseed plant. This gene, what kind of sequence or protein kind of sequence it can able to provide. Fine. So for that XPassay, you need to open. This is the URL for XPassay. So you need to open the XPassay. This is the homepage of XPassay. So here proteomics, this particular hyperlink will be there. You need to click on this proteomics because you wanted to get the informations with respect to the gene, the protein sequence. So you need to click on the proteomics. So if you are clicking on the proteomics button, and next immediately some few here columns will appear. So from here you need to click on the protein sequence and identification. There will be having protein sequence and identification. You need to click on this protein sequence and identification, and later on you need to click on the uniport KB. Fine. So if you're clicking on this uniport KB, so this is already mentioned, uniport KB. So if you're clicking on the uniport KB, this particular home page will appear. And this home page, this particular dialog box, you need to write the FAD2 gene. Okay. So and then you need to click on the search button. So yeah, I'm writing here FAD2 gene and I'm clicking the search button. So once you're clicking on the search button, you can see many FAD2 gene related information which is coming. Fine. And this is the entry is indicating this is the particular your information related one kind of accession number. Fine. So if you remember this accession number, directly if you're putting here in the query, and if you go for the search, immediately this particular gene information, that sequence only will be getting. Fine. If you do not know this entry number, so that time you need to write directly the gene name, and then you need to go for the search. So I'm talking this particular, I have highlighted because this particular sequence I'm going to click. So once I'm clicking on this particular sequence, this particular hyperlink, so immediately I'm going to get, this is the protein sequence. So the gene, this kind of this FAD2 gene, which is going to produce this kind of particular protein, and the proteins are having, this is a particular amino acid sequence. So if you can click any one of these, So it will be providing you similar type of your this protein sequence. Now here you need to we need to remember one thing, that is what is that? Okay, so here we need to remember one thing, that is when somebody has something happened.
Somebody is scribbling.
Yeah, something, some
Scribbling by using the pen.
Yeah. Oh, so now if we click any one of these, okay, so here you just look at here, so here most probably 380 and another 10 if we add 390, so total 390 amino acid sequences are present. Fine. So these 390 amino acid may not be there in all this particular sequence. So if I am clicking here, might be it will be more than that. If you're clicking here, it might be 5 or 10 less than that, because some who has submitted that particular information, how long they have considered the particular gene sequence. Based on that, they have got the gene related information and they have stored here, and based on their submitted gene related information, the software is going to analyze the protein encoding part. So software does not have that capacity that this is the best one or this is the best one. No, software does not have in this aspect that particular capacity. Software just providing all those particular information. Now you need to understand which one you are going to use for your own benefit. So that one you need to get, and look at here. Once we are getting this information, here once again you can look at here, the blast option is given along with your pairwise multiple sequence alignment, like many word will come here, you will come to know later one. So if you wanted to do the blast with that, the sequence I'm having, just I'll be clicking on the go, I'll be doing the blast activity with this particular sequence. And if you want to do the multiple sequence alignment, the options also will be here. So we can go for again, we can click on the go, and lot of multiple sequence alignment related informations and result will be appear. And with respect to the phylogenetic tree also I am going to get. So here just I'm giving the idea, so how we can extract the protein informations. Either you can get it from NCBI directly, no problem, or else if you're using the XPassay, so you can go this way, we can get the many similar type of gene which is going to encode the protein, that information, and the protein whatever you are getting here, you can directly go for the first blast and all other activity. So this is that the protein and nucleotide sequence always is required for us for doing our next stage activity. So here I just explained that how we are retrieving the information. Now based on your aim of the research, you need to use that retrieved information for comparison purposes, for knowing the structural activity of your gene or the protein sequence, to do modeling with your protein sequence, or ultimately we can go for drug designing and all other kind of activity you can able to do with that. So with that I'm here I'm completing. So if having doubt, any queries, please you can ask me.
Participants, if you have any doubt, you can ask regarding the retrieval of sequences or nuclear, all those.
I just one thing made minor, just if nobody is having any doubts, so I just go for the made minor. So that part is left out. Just it will take another 5 to 6 minutes or hardly 7 to 8 minutes. Is it okay, Amjad sir?
Yes, sir. Go ahead. No problem.
Okay. So if you are having any questions, kindly ask me. I think nobody is having question. So I just want to show the med miner how to use.
Yesterday, Deepankar was asking about data mining. I hope he's not. Deepankar, you have any doubt regarding the data mining?
No, my question was like that. due to huge amount of next-gen sequencing, huge amount of biological data is generated. How we can use this huge amount of data, that is big data, through data mining for bioinformatics analysis. My question was like that.
Yeah, already data mining, I mentioned how it is actually working. What are the different layer it is there, and based on the layer, we are extracting the information. Fine. So if you are expecting that 1,000 or 10,000 or 10 billions sequence, whatever deposited in the database, at a time I can able to compare with everybody, it is not possible, because so far there is no particular type of software which can able to analyze at a time all the 10 millions or billions type of base pair. It is all about depending on what kind of keywords you are providing, and based on the keyword you can select. You can select that this 50, 60 or 100 I want to compare, I can get the information from them. That is possible, but it's not like billions or trillions base pair at a time. It is never possible.
Thank you.
And if it is a microarray, I come to the next point. If it is a microarray, that is microarray only, it is giving the huge information which related to all kind of gene, right? So now it is feasible for us. We can use the microarray data. With the microarray, I can analyze anything, 10,000 or 11,000, whatever the information is there at a stage, I can compare. In the normal software or normal speed, your what is the network, it is possible. It may take 1 hour, 2 hours, 3 hours time, but it is possible.
Okay, thank you.
Welcome.
Participants, anybody else?
I think MedMiner, sir.
Yes, sir. Yes, sir. You can go to MedMiner.
I want to use the net here directly, Chrome.
Yes.
So
yes,
shall I minimize it and yes?
Yes, yes, you can do it, sir.
No, I'll be minimizing it, then I'll be open separately Chrome, or how I'll go actually?
So your screen is visible, sir. You can.
Okay. Then I can go directly Google Chrome.
Directly, yeah.
Yes. Somebody has snatched it. Is
they only have to arrange it.
Okay, so my MedMiner directly need to type in the Chrome. Med. So MedMiner basically an internet text mining tool for biomedical research. So MedMiner useful for biomedical research. You can get various kind of research article basically here. Okay, so all maximum will be in the BMC journal connected articles, and this is basically useful for medical purposes, and the doctors who are doing research or doing MD studies on the various particular aspect of physiology. So they basically prefer to use this MedMiner. So MedMiner, an internet text mining tool for biomedical research. So just to type the MedMiner. So it is started from 1999. Okay, so it is not too much very old. And here full text PDF, you are going to get it. And every 1990, this was the first that paper, whatever they have uploaded. So every time if you are opening this MedMiner, they are going to project that first particular publication here. Okay, and you can look at here years are mentioned 2011, 12, 13, 14, like that. If we go scroll down, you can see many year 1990 first they have projected. Always it will be coming in the 1919, and then 2000, 2001, like that. So you can if you wanted to get information 2020, so you just click over here 2020. So these are the different informations which is directly related to the your medical field or biomedical research. You can get it here, and you can see these are all are hyperlinked. So you just click on this hyperlink, you can able to access the full paper. Fine. So you can able to download this full paper. As I mentioned already, BMC journal it will be. So these are all authenticated, and I repeat, for MD purposes, doctors basically use this particular site. And if you do not, if you are doing research on cancer or certain epigenetics or any particular aspect, so you can also get literature from this particular database, and it is very much reliable, very much authenticated. Suppose if you are clicking on 2017, so 2017 in the BMC journal, these are the particular paper has come. So you can able to download this paper, no problem. And if you click on the tools, so these are the tools are available. Okay, cell miner if you are clicking, so cell related aspect, whatever the information on the paper basically is having, they are going to provide you. Okay, so you can, if you wanted to get the mRNA mean values or whatever is this, you can select from here. You can able to select whatever you are getting here, you are fixing. Okay, so and you can choose file, you can upload your file. This is actually, if you are publishing a very good paper, you can upload directly here. And of course they will be analyzing your paper, whether the paper is having DOI number, etc., not, and which particular the paper is, suppose microRNA something, or it is related to the complementary DNA related some informations, or if it is related to the some kind of gene transcript related information, or my pattern expression related informations. So you need to click it here, and you can upload the file over here. They can analyze it. And query genomic data, here the genomic data, if you are having that accession number, you can directly give it here. Otherwise, directly gene name itself, you can type over here. Fine, as the example is provided here, ABCBP1, BRCA2, etc. So you can directly type here, and you can click on the get data, and you can provide in each case your email ID. So if you are offline also, if you are searching for you, they'll be sending that information to your mail ID. Like query drug data, so whatever the drug related information you want to get, this is sometimes help us to know that what are the particular drugs for what purposes. It is already there in the market. So similar type of drug component for designing and developing, sometimes we can use this kind of database. So here, what kind of drug, like the drug will be having cytidine, or if you based on the molecular weight range also, you can filter them, or based on the molecular formula, you can filter them. So you need to write the particular drug's name, okay, or accession number, etc. If you know, according to the drug data bank, whatever the number is there, if you know, directly you can type, and you can click on the get data. So we can, many things you can download. Okay, I'm not going in this part again. So you just this could be your practical. So you can type, and you can try to find out that all informations related to that. Again, I'm going to the home page. So this is the home page where it is on. Not going back. Okay, I'm opening again. How to minimize it? Yeah. So cell miner, so already mentioned cell miner, like you can move whatever you're looking for. So this is the your index. So all this kind of activity or information you can gather from here. But we basically use to download those particular paper, which will be useful for our research activity. As I've already mentioned, this is for biomedical research. So somebody is genome related and all kind of activity they're doing, and they can use basically this kind of database. So you can try with your own, and if you are having any doubt, so we can discuss in the practical session. So this is the way, just basically MedMiner we are using, and you can see here National Cancer Institute Center, again that is connected with the NIH, National Institute of Health. So it is from USA, they are maintaining this particular server. So as I mentioned, whatever the paper, whatever the things we are submitting, they'll be scrutinized very properly. And if they found, yes, it is really new informations you are going to present, so then only they'll be accepting and they'll be putting your paper here. Otherwise, they'll be just rejecting. So it is very better way to get the very particular authenticated literature with respect to your biological research. Thank you.
Any doubts, please? You can ask with DIPTENDU sir regarding MedMiner or PubMed or retrieval of nucleic acids and protein databases or anything.
Yeah, kindly, if you are having a doubt, please.
You have finished yesterday the literature.
Hello, sir.
Yes.
Yeah, Mahesh here.
Yeah,
just
Mahesh,
wanted to know how to retrieve a specific information. You did tell me that so all the papers which are arranged chronologically that can be retrieved from the database which is already there. So, for example, I would like to retrieve data from COVID. How do I do? So, is there any search engine which could retrieve a specific information, or is it just like already published papers could be downloaded?
Yeah, Mahesh sir. Actually, if you are now opening the NCBI, there itself they are giving separately hyperlink one all COVID-19 related information.
No, from NCBI, I know. From the minor thing that you told me, the last database that you
from MedMiner, I don't think so. It will be available. Basically, they are giving emphasis on all kind of cancer research.
Okay.
So I mentioned it is biomedical research. So,
so only the published one, so which is already there, which is already been published, I can retrieve. So not a specific one.
No, not the specific one.
Okay.
From there, it is not possible, sir.
Okay, fine, sir. Great. Thank you.
And WHO COVID-19 one that web-based one software they have created. So we can get.
So I was like trying to download the sequences of COVID. So the nucleic acid sequences are not available as such. So the genomic sequences. So only the proteomic part is available. For downloading the genomic sequences, Uniprot I used. Okay, for proteomic part. But for nucleic acid sequences, I don't think it is much of.
It is not much is available, right, sir?
Okay. Fine. I'll discuss it.
If some literature, if they have mentioned some accession number, you can
from the accession number I can retrieve it.
Other than that, whole genome information till now.
And for the mutational analysis, is there any specific software which could?
Yes, it is there, sir. It is not only single software. Actually, 5 to 6 different web-based free software is only.
I think.
Entire genomic data, it is really difficult because whatever
huge amount of data,
open-based server I am telling, none of it is using that huge information. They are restricting with the 500 or 100 or maximum 1000.
Exactly. Yeah. Fine, sir. Thank you, sir.
Welcome.
Okay. So good morning.
Good morning, sir.
Yesterday we just started discussing about the flow, what are the experiments that we will be covering, and today I just thought of starting with the first database, and even in the previous speaker also explained about the NCBI and other databases like Uniprot, Swissprot, and he also showed the MedMiner aspects. So it may be a kind of repetition, so I will not go into the details, but just thought of showing you where and all it is present and how exactly it typically looks like. So this is the NCBI, and since it is kind of a practical session for the next 2 weeks, and based on looking into the different participants, right? So one of the requests is you don't need to feel like there are too many terms or to store how much of this information in your mind. But it is better if you just get a feeling that this kind of a data is available. Okay, if you get that much, that is enough. Then when you really want it, you can always search in the Google, and then you can find the database name. But if we are working out towards making you to aware that such data is available, because I can easily make out that there are many participants from different backgrounds. So you feel like there are too many things suddenly happening, right? Database names, server names, algorithm names, everything. So in that particular context, NCBI, if you remember, that's more than enough. So if you can remember, like National Center for Biotechnology Information, that's well and good, but NCBI is more than enough, because this B, biotechnology, will vary from one database to another database. So if it is EMBL, that there it becomes like biology, right? So not the biotechnology. DDBJ, DNA Data Bank of Japan, so there it becomes like data bank. So at DIPTENDU, protein data bank, again B becomes bank. So there are small changes, but don't worry about all those things. Just remember, NCBI is there, we have all the related information. So created in 1988 by the NLM at NIH, and these are the purposes why they started. Establish public database, research in computational biology, developed multiple software tools, and then to disseminate the biomedical information. So this is how we will work out with this one, the NCBI and all other aspects. And then the next second database, which we will also look into, maybe today or at appropriate times when we are doing the work experiments, we will use one or the other databases in a different manner. So another database, which is EMBL, European Molecular Biology Laboratory. So Heidelberg is the main station or main laboratory, and these are called as the outstations: EMBL Grenoble or EMBL Monterey Tondo, EMBL EBI or EMBL Hamburg. So I was lucky enough to work at EMBL Hamburg for 9 months, EMBL Grenoble for 4 months, and then in both the times I could visit EMBL Heidelberg. In both the times, 2 times I could visit here. This is the training center, basically whatever the building is there we could get the training of specific aspects. Then in 2014 I could go to EMBL EBI Kingston again for 1 week practical exposure, and then learn some of the aspects related to structural biology or bioinformatics. So not with Monterey Tondo, Italy. But yes, both the things, if you look into, this is structural biology and this is also structural biology. So some of the crystallization work and structure determination work is going to happen there, and bioinformatics aspects will be done in the Kingston. So this is how the Kingston building looks like, which is there in the Welcome Genome campus. And this is how the website of the EMBL looks. Again, we can do a lot of aspects. There are so many resources available, so many tools available. They have prepared some servers. Training is also happening, so one can look into these training aspects. Now most of the aspects are training is virtual, so one can register and then take part in the training also. So it is not restricted, okay, only with the bioinformatics or structural biology. Here in the Heidelberg, it's general. So molecular biological aspects or plant related aspects. So all different types of the research is going to happen there. Now this particular slide I put intentionally so that you will get familiar with the terms, because even in the yesterday's presentation and even in the today's presentation, you keep hearing the terms Like NCBI, we say EMBL, then VDDBJ, EMBL. So how are they connected as a holistic view? If we look into, are they connected or not? Are they working independently, or how exactly? So as yesterday also mentioned, the databases are maintained. Okay, the rationale why we have like 3 separate databases like GenBank, EMBL, and DDBJ. So to address this question, first thing is US people thought they will store their data in their database, which is there in the National Institute of Health in US, that is Bethesda. Europeans said like we will store our data only in the EMBL. We will have our own database. Then the Asians said like we will store the data in the Japan under DDBJ in Mishima, Japan. Then over a period of time, they thought like the people who get the sequencing done in EMBL, again the same sequencing used to be done by some groups in the US. So it's a duplicate basically. Then the consortium came into existence. They said like this is not the right way. If you deposit one data, again someone else will sequence the same thing and deposit into EMBL, and someone in the Asia will also do the same sequencing and then deposit here. So it becomes like a triplicate. And then once we know like they have deposited here, this guy can also reproduce the same thing and deposit here and claim like a new one. So to avoid all these things, so this consortium, that is the INSDC, the International Nucleotide Sequence Database Collaboration. So they created and said like, we don't need to sequence again once it gets deposited either in GenBank or in EMBL or in DDBJ. We will share the data across all 3 databases on a daily basis. Okay, so on a everyday basis, if we let's say if we sequence and deposit into DDBJ, so the next day the same sequence will be available even in the GenBank and also in the EMBL, so that they made it like a integration. So these are the places where we will have the data now. GenBank is a database maintained by the NCBI, and it is there at National Institute of Health, Bethesda, USA. EMBL is once again a database which is maintained by EBI, European Bioinformatics Institute, which is further present in the under the EMBL. In the same way, DDBJ is maintained by Center Institute, and which is further maintained by National Institute of Genetics. This is Center for Information Biology, and then NIG, National Institute of Genetics, Mishima, Japan. Now, I want to search all the databases, because if we look into NCBI, we have 100 plus databases. Now, how should I search this database? Then they made their own search engine, just like we use Google and then search. So to search the databases, whatever is there within the NCBI, they created their own search engine that is referred as Entrace. EMBL created their own search engine, referred it as SRS, that is Sequence Retrieval System. Then the DDBJ created their own search engine, and then they called it as Get Entry. Okay, then you can submit, you can update here, people can submit and update here, people can submit and update here. So if you can remember this particular slide, okay, most of your confusions won't be there. Now, it is only the sequence that will be shared on a daily basis. Okay, so the services provided by GenBank is different. Okay, services provided by EMBL will be different. Services provided by DDBJ will be different. Since these 3 are anybody, whether it is a primary database or secondary database or a specialized database, GenBank, EMBL, DDBJ.
Primary database.
Yes, so all 3 are the primary database. Why we call it as a primary database? The moment scientist does the sequencing, the sequence, whatever he gets, they will just deposit into DDBJ or EMBL or GenBank. Then comes the analysis. So we need different tools for the analysis. So EMBL has built some specific tools. GenBank has generated or made some specific tools. DDBJ is also made some specific tools. So keeping this in mind, okay, with this particular background, I just thought of starting with the NCBI. If you are doing parallelly, you can also start working. And if I am going a bit fast, you can also let me know, like I can just go a bit slow, okay, so that we all will be in the same page. So now you can open this NCBI homepage. And see, when you are a beginner, you don't need to remember like ncbi.nlm.nh. Just type even in the Google. If you just type in CBI, it's more than enough. It will take you to the homepage of NCBI. Now, once we have this, now within this NCBI, as I mentioned, they have so many other databases that are there. Now, to avoid searching all these databases by giving a keyword, they have created the Entrace. So Entrace is the search engine that works in the background. Now, whatever the protein of our interest or anything, we can just type here. Now, as I mentioned in the yesterday, my plan is to, at the end of few experiments, or at least 10 experiments, I want to use one protein, and in each experiment or each separate database, when we search, we will use the same protein to get to know like the kind of information we are going to get. Now, one of the protein that I thought of working is human CCR5. Okay. Now, as in the morning presentation and also in the yesterday's presentation, people have already mentioned about the PubMed. Maybe I will give you 2 or 3 minutes time. Okay, just tell me what is the role of this particular protein. Maybe you can spend some time. Just check out. Okay, what is the function of this particular protein? And then maybe you can raise the hand and then you can unmute yourself. Then you can tell me what is the role of this particular protein. So organism is human, protein is CCR5. Yes, Arindam, maybe you can unmute.
Sir, it is a co-receptor.
Yes.
During entry of HIV virus.
Correct. Perfect. So from where you obtained this data?
Yes, sir.
From where? Like any particular article?
No, sir. During studying AIDS, pathology of AIDS disease, then I came to know this.
Correct. So not with the search, by default, like when you are in your course itself, you got to know about this particular protein, right?
Yes.
Okay, good. Now, what I will do is, I will tell you 2 aspects, okay, as a systematic search to understand, and also another one is non-systematic way of understanding. Then how should we go about understanding any of the disease or any of the work that you want to do? Now, I will tell you the challenges associated and why we go for non-systematic method. Okay. Now, as per the overall discussion that we made since yesterday, right? What I did here is I have selected all databases. Then I will type like human CCR5 and then I am going to search. Now what it is doing is, it is searching all 29 databases or whatever the databases that are there in the NCBI using Entrace. Then it is going to show like there are 29 results. Okay, results found in 29 databases, and these are those 29 databases. Now we already know that in the literature, nothing but PubMed. In PubMed, we have around 8,637 articles. In the PubMed Central, we have around 35,000 and a half articles. Genome related. So the classification of the databases are also made, like literature, bookshelf, reading. So it's related to literature like that. Gene related, gene expression omnibus or homologene. So all these databases are the information is related to genes. All these databases are giving the information related to proteins. Protein, identical protein groups, structure, information of the protein, conserved domains. If we want the genome information, all the genome related databases are placed here. There is no data related to assembly, there is no data related to biocollection, but yes, genome information, yes, there are 11 entries, okay, which are related to genome. Clinical trial information or mutation information, DBSNP, variation, OMIM, online Mendelian inheritance in man, entries, structural information, there are bioassay information. So as a one shot, okay, we will get the overview of the kind of a information, that is the frequency or quantum of information that is available related to this protein, we will get here. So if we look into this, we can easily make out like there are many papers available. 8,500 is quite a good number. That means there are many who are working on this particular protein. Gene is well characterized, domain is well characterized, protein structure is also available. If the protein structure, if it is 0, that means the structure is not available in the database. So that means the determination of the structure for human CCR5 is not yet done. So that's the kind, it's a complete overview of the protein you are going to get. If we remove off this human and search, right, then the CCR5 that is present in other organisms are also we are asking to search. So now if we look into, it has increased from 29 to 32 databases. Then we also have more entries everywhere. It has gone from 8,500 to 10,000. We were not having anything like compounds. Here everything was 0. Now we got like 11, right? So this gives how specifically you are going to search, okay? And you more specific your search is, better will be your data curation, okay? Now one of the tasks that you can do is, as a first experiment, we will go for this PubMed, okay? And then when I say like Homo sapiens or human, okay, we have around 8,704 results. And for any of the project or for to do the research, going through all these 8,500 articles is impossible. We may not be able to do. Now from this stage onwards, and since for many this human CCR5 may appear to be a new protein or a new term, one can do the another method which is not a systematic method, but that provides a information about what exactly it is. You just type human CCR5 in the Google, okay? Then if you want, you can go to Wikipedia, okay? People do say like one should not refer the Wikipedia for the research and other aspects. But as I mentioned, this is a non-systematic method, okay, or not a systematic method, but you will get to know what it is at the first instance. So rather than being to be like completely unknown, at least get known with some aspects. Now here also, as we mentioned, right, it acts like a co-receptor for the chemokine. Then it codes for the CCR5 protein, where it is present, chromosome location, and all those information. It is involved in the HIV-1 infection, its structure, its accession number, right? So hyperlinks. But what is important, why people say like don't use Wikipedia is one can edit the data. Okay, that's the only concern why people say like don't use the Wikipedia. Now, another major question, maybe someone can answer it now. Why should we go only with the PubMed? Because even yesterday and today morning also people mentioned like we should use the PubMed for our research. But looking into these, right, frankly speaking, we will not get to know anything about the protein. Right, where it is mentioned like co-receptor for HIV, it's too difficult to understand. Even if you read any article, still it is too difficult to understand because the paper itself writing is in a complex manner. But look at here, it's very straightforward, easy to understand. And we also have references given on what basis they say like it is resistant, all these things. We have references and we can easily understand. First thing is understanding the protein. Then our hypothesis testing and all those things will come. So spend some time in Wikipedia or anything, okay, and understand the protein, whatever you are interested in. But we will not cite the Wikipedia. It is something like we will use, but we will not acknowledge them. It's the kind of a problem for the Wikipedia. Right, even in any of the programs or any of the functions or anywhere, there will be a person who contributes a lot, but we may not be able to acknowledge them because of authenticity, right? So what authenticity is there in PubMed which makes people to say like use PubMed? Any guess or anyone? I hope the question is clear. Or do I need to repeat the question? Anyone?
Could you please repeat this, sir?
So the question is, you may be aware that don't use Google or don't use Wikipedia in the references whenever we write an article. But we will go for citing the article which is there in the PubMed. Now, our people also say, even in the morning talk or yesterday's presentation, literature search means use PubMed, right? Now, what made people to say use PubMed, not to use Google? Why?
Because the publications in PubMed are more authentic because these have been reviewed properly.
Yeah. Now, can you tell me the criteria to say some data is authentic?
That means this should be reproducible.
Okay.
And other people doing the same type of research will get same type of results.
Okay. What if they don't get the same result?
Then it should be statistically proven. That means,
right? Someone else, please make an attempt. It's only 4 parameters, okay, that we need to say something is authentic. When we can say like, now I will put it into a very simple terms. What is the difference between the published article and a blog? You know the blogs, right? People do write some blogs. Now, what is the difference?
Personal opinion, and it is not reviewed.
Correct.
But in case of scientific journal, high the index journal, it is reviewed, it is criticized. Sometimes it is accepted, sometimes it is not, sometimes it is said to improve.
Yes,
In case
sir.
of published research journals, well, good journals. But in blog, anybody can write down anything as per his or her opinion. It is not reviewed.
Correct. Perfect. That's the major difference. So the review is going to happen in the journal. And now the four important aspects when we say the data is authentic is, the first one is we need to know who worked. The name of the author is most important. Let me take any example. Okay, just the first one. Now, if I expand, right, one can write a blog just by mere mentioning the name. Okay, but whether by getting a name, can we call the data as authentic? No, because we need the affiliation also. If a scientist is there and affiliated to this institute, okay, even if this guy leaves this lab, the workbook or the logbook will be there here. Okay, so the data, if I want to reproduce, and if I'm not getting the same result, in this paper he would have given the methodology and I worked in the same methodology, but I did not get the result. Now in the case of blog, we do not know who is that guy, because the same name, Chao Wang, maybe there are hundreds of people in the China. Who is that person? Now we need this. We need the affiliation. Let us say Prashantha Karunakar. I have written a blog and I said, like, if you do these steps, this is the output and this is going to cure the disease. Now, if my affiliation is not there, right, there are many Prashantha Karunakars that are there. It is not only me. So the second question is the name. What if only the affiliation is there, not the name? That is also not useful, because if they come to Department of Biotechnology in PES University and ask, like, we got this kind of a work done, but department will say, like, we do not know who worked on this, so tell me the name. So the name should be there, affiliation should be there, and when they have worked, year when they have worked, that is also important, because the claim saying, like, I have developed a drug against CCR5, but if the work is done 20 years back, then again it is of no use, because the protein would have gone for a mutation or whatever. When the date becomes important. Then the third one is it like you are self-claiming that you have developed a drug, or is there any community who says that yes, whatever you have identified can be made it public to say that you have worked or your results are accurate? So that process is nothing but peer review. So I will send my results to a community, that is nothing but this journal. There are set of scientists who will review the work, maybe 3 iterations, 4 iterations, or 5 iterations. If they are not convinced, that means this community, if set of reviewers belongs to this journal of medicinal chemistry, if they are not convinced with my work, then they will reply back saying, like, reject this work. I will get the rejection. Now what I will do, I will get to know, like, okay, this community is not happy with the work, or they are not able to understand because my approach is different than what their expertise is. I will communicate to another journal. Then that set of community scientists will find it okay, novel, and then they may accept. Then the year of publication will also be there. So to July 15, 2021. Now if the date is not there, if the author name is not there, or if the affiliation is not there, or if the journal name is not there, even if a single parameter is going to miss, then we cannot claim or we cannot say the data is authentic. Now, why people say, like, don't use Wikipedia then? Because there are many sentences in the Wikipedia where we may not be able to find a reference. So when we cannot find a reference, then we cannot propose anything. If I am going to develop some drug for example, we have a situation, let's say, for example, this. Here it says like CCR5 is the primary co-receptor used by GP120 subfunctionally with CD4. But for the throughout paragraph, the reference is given over here. But whether this reference should be used for the entire paragraph, or what if after the full stop, only for this statement this reference is cited, not for this, then we have a problem. And after your hypothesis and let's say 2 years of research, and if someone says like this GP120 and CCR5, they are not connected, then if you say like this is where it is present, I read in this particular, then they will say like even I can also edit. Okay, even I can also edit, say, then rather than CD4, I can write like CD5, right? So that is where the problem is. The problem is we do not know who wrote this particular sentence. Who wrote this particular sentence is not available in the Wikipedia. So go for this. Now every article that we go through, check when we read the abstract, and if this particular information is useful. For example, randomly I'm just telling, yes, who has written this particular statement? I have information. These are the guys who written. Where do they belong to? They belong to this. When did they write? They have written and published in this particular time. Where did they publish? This is the journal where they published. I have clear information. And if this is, and if I work out, and if I am not going to get the results, catch hold of this guy. Okay, you have a person to check with. You only proposed, I am reproducing exactly in the same manner, not getting the results. Can you tell me where I made a mistake? Right? So we need to make research responsible, and when we are planning something, we are taking some others' work as a reference, and then we are designing our work. So we need a responsible people results to be convinced or to be considered for my work. So that is where this information becomes extremely important. So in future, when you are working, if you do not find any of these information, so it's better not to take that particular information, as long as you are doing the especially in the research. Okay, that is why in most of the journals also, in the reference section, when we write, they ask for this PubMed ID. So if the PubMed ID is not given, so they will say like that particular reference is not valid. Okay, that's how it goes. Now to continue with this, now again, as I mentioned, you can go through this and get a feeling of what exactly this particular protein is. But as I mentioned, human CCR5, if I type, if I am going to get like 8,500 articles, it's impossible to read all 8,500 articles. On what basis it is giving this 8,700 is if the term human and CCR5 is there in this, it is going to fetch that information and it is going to show like these many entries are there. Now you can spend again 5 minutes, okay, can just quickly understand this particular protein, okay, and then add the keywords and reduce this number of hits from 8,704 to maybe like 70. Okay, now to add another keyword here, right, if I want to type like antagonist, for example, now from where I got to know like I had to type this antagonist, right? So that comes after reading this, because whether I should type agonist or antagonist, whether structural information, whatever. So if I quickly go through this, I will get to know like, okay, this protein is responsible for HIV. So I need to have a drug molecule so that it will stop the activity of this particular protein. So to stop the activity of the protein, I need a drug molecule, right? So then the term drug will come. And then if I search, how many are there? Still 3,000 is there. Can I read 3,000 articles? No, not possible. That means I still need to filter it out by giving additional keyword. So these are the keywords that we are going to give to reduce the count.
Sir, with inverted comma, it is reduced up to 143.
Yes, sir. So what is the keyword that you have given?
Human CCR5 with inverted comma.
With
Inverted comma.
inverted comma.
It is reduced up to 143.
Yes. Now, my most important concern is, though in the previous presentation, it was given like author's name, all those things, right? Flower brackets to exclude. So you don't need to remember any of those things, okay? Giving the author's name or other stuffs. For us, final importance is the project, whatever I am going to do. So my intention, if it is to develop a drug molecule against the CCR5, and the PubMed or whatever these things have developed, right, they have done it in a very sophisticated manner. So even if a layman types, it will take the synonyms and it will fetch all the information and gives a result. So my keywords should be in a way that it should match to my idea of working on this particular disease. So in that particular sense, give a keyword where you can type a word which should match to your idea, whatever you want to do. So that means if whatever the keyword, that's why I'm not specifying anything. So any plant-based, if you want, so just check, is there any plant-based drug against the human CCR5? Again, so we got something like 6 now. That means there are few papers which tells about plant-related drugs are there to control this human CCR5. 6 is good enough, so we can easily read. Like this, you keep changing the keywords based on your interest. So the interest may be structural related, drug related, or gene related, domain related. It will vary. That is why we cannot restrict. So anything of your interest, you please type that. If somebody is interested only in the mutations, whether the protein is undergoing any mutations or not. So mutations, yes, there are 1400 results. Then again, if you look into some aspects of this, so that is why the literature search or literature review is a tedious process. Okay, it's not a easy method, and the way you search with the same concept and your friend searches for the same concept, the article that you are going to find will be different, because the keyword varies from one person to another person and the tool works based on the keyword. So how good you are in searching, that plays extremely important role. So let us say this protein undergoes some mutation and causes the disease, and there is something like CCR5 delta also. Now automatically if I put like delta, it comes down like 101 results. Okay, so what you want basically in that particular order, if you keep adding the keywords, you will get into a specific aspects. Okay, that's the overall rule. If I want to put this one into a general perspective, I want to know about the Bangalore, or let's say any city, Chennai, type the Bangalore, then within Bangalore what I want to know about Banashankari, then within Banashankari I want to know specifically about Huskerihalli or PES, and then within that whatever I want, you type there. So at each level, biotechnology. So you are going in depth. If I just type like Bangalore, we will have thousands. Bangalore with PES, again there are some. Bangalore with biotechnology, again there are few. But within this specifically, you type one more term. Within biotechnology department, you want some aspect of the biotechnology research, whatever is happening. Then you type that. So you are filtering in a way that you want only the specific articles of your interest. Okay. Even we do the same thing, okay, even in the Google also, so that we keep adding specific keywords to narrow it down. That's the only way by which we can narrow. Once we do this, okay, so the next level we go for. So one can select the articles of your interest. Okay, what I usually recommend is quickly go through the abstract. Okay, and when you go through the abstract, you can easily make out whether you need to read the full length paper or not. In most of the cases, from the title itself, we should be able to identify. But in few journals, they restrict with the length of the title. Okay, so in such situation, we may not be able to understand it in detail, what exactly they want to convey. Then you go through the abstract. Then if you find something interesting in the abstract, then you look for the full length paper. Then you go for this PMC, PubMed Central, then read the entire paper. Because if you start reading the entire paper from the beginning itself, right, we may not be able to cover all 101 articles, or we may not be able to get we may miss out few important papers. So that is where the search engine or the search terms plays an important role. Any questions, sir, or any doubts with anyone?
Sir,
Yes.
the same, I have put the same keyword human CCR5 mutations delta. But the paper that retrieved, first one shows the title evolutionary views of the AIDS process.
Okay.
But you got different paper. What might be the reason?
No, the number of articles will be the same, but it will look into the history. Okay, so now people have used this machine learning and artificial intelligence everywhere, sir. So the results will vary. Okay, but the papers will be there, may not be in the top order, but will be there in the second or third order.
Yes, sir.
Yeah,
same number of papers are there,
yes.
Only the sequences are different.
Yes.
Okay, sir.
Because any time I search, right, there will be a history created and it will be stored within the PubMed. So to my laptop, the history will be stored and it will check what all I searched and what is that I'm looking for. Then it attempts to show the more relevance results here. So even if I type, let us say here, if I type like Prashantha Karunakar, the results, whatever I'm going to get, for example, in the Google itself, whatever the results I'm going to get here and whatever the results you are going to get in your computer will be different. Because the search engine is so optimized that it works to give the best result whatever I looked earlier in the first hit. But for you, it was going to be different because it's a fresh search, first time search that is happening in your computer. So the results will vary. But these things will be there, but not as a first Google Scholar, second the faculty, then my site. It may be of a different order, but it will be there.
Okay, sir.
Yeah. Now, once we have these abstracts, you can go through. Then I prefer to write the abstract in your own ways or in own words. Okay, let us say after doing the search and finding out the 2 or 3 important articles of your interest, right, you can just highlight few aspects or copy paste it into a Word document and then make the information ready. One can also search based on the author's name. Okay, you can just type the author's name in the PubMed and then you can say search. So there also it is going to give the articles that are there. So this option of searching based on an author's name is useful when you are sure that this is the person who is working only on this particular area. Okay, otherwise it is not recommended to type the author's name in the PubMed, because we do not know who is working. But when you are sure that only this person is working, okay, on the research of your interest, then you type the author's name. Then you can also filter it out based on let's say I am working with the febrile disease. Then you search like how many are there. This gives information like how many papers this particular author has published related to this disease. So there are 2 papers. That means he has published 2 papers related to febrile, whether this author has published. So you will get to know like what is his interest or what are the different diseases he is majorly working with. So if I type the author's name and then cancer, okay, there are 2 papers related to cancer where this particular author has worked with. So diversity you will get to know. That's the advantage. But when you are doing for your research, don't restrict with the author, restrict with the concept. Okay, restrict specifically with the concept. What do you want to prove? Now, once we use this PubMed, right, we can, as I mentioned, we have different options. We can send the references, bibliographies, and then you can export it out. There are different ways by which we can export. That's the second option or secondary thing. First thing is to know, okay, we can create a file and then we can always open that in let me save this, and then we can open in the Notepad++. Then there also we will get some citation information. Okay, so the publisher name and the where it is published, the title of the paper, abstract, people who are all worked. Like this we will have for 2 articles, complete information. Anyway, those things I will do it like that. Is once you have written the paper, then the citation, one can use the Mendeley or any other tools. Now let us, any doubts with the PubMed, sir, or PubMed with anyone? Anything to be done in specific or any queries? Okay, so if nothing is there, now we will go to the another important thing, that is the gen bank, or also referred as nucleotide. Okay, now we know how to retrieve the published articles, right? That's what we did now. Now I want to retrieve the sequence for this human CCR5. Then again you can type like human CCR5, select nucleotide, then say search. Let me close this. So just to give you some hint, this is how the protein looks like, CCR5. This is a 7 transmembrane helix, and this is the lipid bilayer surrounding the whatever is there. Okay, maybe in the yellow color, 1, 2, 3, 4, 5, 6, 7. There are 7 helices. Helical representation is like this. Then this is referred as a extracellular, and this is referred as intracellular region. HIV binds to this, and then it enters inside the body. So first it binds to CD4, then it binds to CCR5, then enters our body. Since it binds to CD4 first, just like not able to see my face. Is there a way by which I can see whether in the video I am covered or not? It's something like, I don't know, first it binds like this, CD4, then binds to CCR5, and then enters inside. That is why this CCR5 is referred as a co-receptor. Okay, so CD4 is the main receptor. Now, if I type like human CCR5, okay, first thing you need to observe is we have around 11,062 entries in the GenBank. Okay, as I mentioned, GenBank is a primary database, and in the primary database, just want to see the is the camera on, sir? You are able to see?
Yes, sir. Yes, sir.
But I am not able to see my face in any of the means here. So just to check whether the coverage is proper or not. But anyway.
It's clear, sir. No problem. You can go ahead.
Okay, no problem. Now, when you type like human CCR5, okay, in the, as we know, GenBank or NCBI nucleotide is a primary database. Now, when I say primary database, we know that whoever performs the sequencing, they will deposit that particular sequence into NCBI GenBank. Now, how many have deposited like that? There are 11,000 people who deposited. Now we will have like delta 24 allele, we have mutant allele, we have complete CDS, we have covering thousands from the mouse CCR5, and then human CCR5 having 1086 base pairs, 3383 base pairs, right? In both the titles, if you observe, right? mRNA complete CDS, gene complete CDS, CCR5 with truncated isoforms. So many variations are there, different variants. So people have done different kinds of experiments, and whatever here, glycine to arginine at 301 position mutation sequence is deposited. Here, threonine to alanine 239 position mutation information is deposited. So there is a change in each entry. Okay, it is not like the entries are duplicates. Duplicate entries will not be there in the GenBank. There will be at least one character difference will be there, and all such information is deposited into GenBank. Is it good or bad? Will be the next question, right? Any guess? Is it good if I type like human CCR5 and get around 11,000 entries? What is your opinion? Is it good to have like this or should not be there like this? And when it is good and when it is bad? I type something, and when I am searching, I am getting so many results. Now, is it a good way to get so many options or so many results, or is it a bad specific with the human and the CCR5?
So it's good when I have no choice. I can choose from any topic related to CCR5. That means initially I am not specific.
Yes.
In that case, it is good.
Yes. When it is bad, sir?
It is bad when I have already chosen a specific topic on CCR5.
True. Correct.
Yes.
So it is good, okay, when I want to work with a variant, okay, and the variant information, this is the mutation, glycine to arginine at 301 position mutation. Now if I want to work with the mutant, and rather than me to make the mutation in the FASTA format file, I am getting the ready-made sequence here. In that particular context, it becomes a good option. But when I am a new, I do not know about this CCR5, but started to work with this CCR5 just to get its sequence and to understand about the CCR5. And if you suddenly give like 11,000 entries and ask to pick any one of your interest and then start, then we are lost. We do not know from where to start. Whether I should take the mRNA sequence or whether I should take the gene sequence, whether I should take these mutants, right? So all these problems will arise. So to avoid this particular problem, okay, what people have done is initially the results were given out like this, and people have raised too many queries, something like you are making the change from this entry. Again, I will come back to this. You are making the change from this entry to this entry to this entry. If we look into these three, for example, the number of base pairs are exactly the same, 1059 base pairs. Only difference is having one base pair where it is creating these variants or the mutations. When you are creating only one letter change from this to this or from this to this, why are you making three separate entries, right? Why you want to create three separate entries where the difference between these three entries is only one letter, right? So we are making us difficult to take or work with. Then people have come up with another database called the RefSeq, that is the reference sequence. Then they said like, okay, don't worry about all this 11,000, whatever is there, let it be there, okay? Because if somebody wants only this, let him take. But if you want to understand about this particular protein, we have curated the data, okay? We have curated. Curation is a set of people will sit, analyze the results, whatever is there for this CCR5, and write it in a single page. Okay, that is what the curation is. To put it into a simple terms, about the Department of Botany, if it is there, different members would have written, maybe 5 members if I take. So 5 faculty members would have written about the Department of Botany in 5 different ways. But if we look into 5 different documents from the 5 faculty, 90% of the data remains the same because they are all writing to the same department. Only 10% of the information may be different. One person may be concentrating on one aspect, another person with another aspect. But overall, 90% will be the same. Then the chairperson or the HOD will take all 5. Then he is going to curate the data. What he is going to do is, okay, give me all 5 documents. Let me look into the 5. Whatever the common is there, let me put as the first. These are the common. Then whatever the differences are there, the key information from the faculty 1, faculty 2, okay, that I will highlight as a separate sections, 1, 2, 3. So what he has done is, the chairperson or the HOD has curated the data of the Department of Botany and made it like a single page write-up, where it provides the information of all 5 pages. Rather than giving all 5 pages, he has made it like a 1 page. So the job, whatever the chairperson has done, is nothing but the curation. So how did he do this curation? He has checked all 5 entries, looked into the similarities, looked into the dissimilarities, which is important, which is again authentic, which can be highlighted, which we should not highlight. So he will take the decisions, then he makes the final 1 page write-up. So exactly in the same manner, rather than Department of Botany, here it is the CCR5. All publications, information, set out, there are hundreds of curators in the NCBI, or even that's a major job role that is there. Even in EMBL, Hingston, annotators and curators are the maximum job that is there. So they will collect the information and then they will check what is that uniqueness in this particular paper. Then they will write down, and then from the different databases they will make the write-up here, location information, right, genomic regions, where and all it is present. So it is here, it is not restricted with the PubMed, okay, gene information, protein sequence information, expression, where and all this particular protein is present. It is expressed in appendix, it is expressed in lymph node, it is there in the spleen, stomach, urinary bladder, where and all the expression is present, structural, bibliographic information. So they have curated and then made it like a single page. And now if you look into, so if we want amino acid change information or HIV1 binding to this GP120, yes, there is a reference. If you refer to this particular article, you know that the envelope gene is there, envelope surface GP120 is there, and from this you can easily understand. Now if we just look into this particular entry, okay, so you can easily understand what all the information, what exactly this protein is, where and all it is present. That information we are going to get from this. Remember, it is not like for all the proteins we will have this RefSec entry. Okay, there are many proteins for which the curation cannot be done because the annotation itself is not proper. So it is something like you cannot write a write-up for all the programs for all the aspects. Okay, there are few programs or few things where the data is very less, wherein you cannot give. There is nothing like a 5-member faculty where they can provide some information for the chairperson to curate. So if the data is very less, right, then you cannot curate. That's the logic. Okay, now if I look into, for example, I will just take with this human CCR5 gene coding as one of the entry, okay, because we will also work out with the same thing in the future. So it clearly tells that I have a complete gene, complete coding sequence information. As even in the morning it is explained, we have the locus, it has 1086 base pairs, it is from the DNA, linear, primates, when they have deposited in 1997, and one version is there. So this section is referred as locus section. So the GenBank is classified into 4 sections, 4 classifications. One is locus. Locus tells the description, version, keyword, or the organism, then the taxonomy. Second section is referred as references. Reference is the second section, which tells us who worked, okay, and because there is a reference, this data becomes authentic. Okay, we have the names, it is hyperlinked with the PubMed. If I go to this PubMed, I know the people's name who worked and their affiliation. Everything is there. When they have worked, 97, right? Where did they publish? Journal. That is why this data becomes authentic. Another set of people worked out. This is the second section, reference. The third section is the features. In the feature section, it tells us what is the organism, what is the genome molecular type used, cross-reference, what type of the cell line they used, and what is the gene name. If it is coding, from where it starts and where it ends. So from the 13th position, the coding region starts, and at 1071, the coding region ends. And when the translation, protein sequence can I get? Yes, this is the corresponding protein sequence. So the feature section tells about the features of this gene or features of this particular entry. What you can understand, what you will get by looking into this, that they are going to tell. If we want to see, right, even in the morning, professor clearly mentioned like each line is going to have like 60 characters. If I want the 58th position, so I know that this is the 58th because the last entry is 70. If somebody asks like what is there in the 33rd position, which nucleotide is present, so this is 10, 20, 30, 31, 32, 33. So this T is present in the 23rd position. So it becomes very easy to locate where it is present. Now from where the coding region is going to start in this, just click the CDS. Okay, it says like 13th position. Yes, this is 10, 11, 12, and then the 13, ATG, which codes for methionine. Yes, codes for methionine. Then the this is the entire coding region, and this is where it is going to end. TGA, the stop codon. So that's the end of this particular coding region. Now we have the amino acid sequence, we have the corresponding nucleotide sequence in both the cases, which is available readily. Now, even in the morning, the presentation, he mentioned like the FASTA format. Now, what exactly this FASTA format refers to? So, if you just click this FASTA, right? Now, this is what the FASTA format refers to. Now, in most of the programs, it is going to accept in the FASTA format. The format goes like this. It starts with a greater than symbol, then one line description, okay, and then the sequence. So there is no gaps, nothing, okay. After this greater than symbol, this is a comment section. This is for our understanding, okay. The tool is not going to take this as a sequence. It will take this as a comment section. It is only for an understanding. It is something like in the Excel, we give the attribute name, header name, column name, because the if we are adding some numbers, right? So the header, it will not take because it is for our reference. In the same sense, this is the comment section or header. Then the sequence, this is the entire sequence. This is the format referred as a FASTA format, which we will use it in our next experiment onwards. Any doubts with the GenBank? Even morning, one professor asked with related to SARS-CoV, right? If we want the complete genome of the coronavirus, right? You don't need, you just go to this NCBI again. Okay, keep everything as a, let us think that we do not have all these specific options. Okay, then just type like coronavirus, and it will search all the entries, okay, all the databases where and all it is present. We want with the genome, right? So go to the genome and get the genome information. So say like genome. So we have 42 entries related to COVID. So you can check even with the plants, okay? So you can just type like neem. So it will only take into consideration the scientific name, Arabidopsis thaliana or anything. So that it will search. You can easily search details. What I have typed is just coronavirus. But what all the other things it has searched? Alpha coronavirus, beta coronavirus, or gamma coronavirus, or coronavirus. So all these. So anything if you type, even if you type like heart attack, for example, okay, if there is a mesh term in the background, medical subject headings. So it will look for related aspects, related terms that one can use, then it will fetch. Because in few cases we may not be able to give a scientific names, right? We are typing like human, but in principle we should not type like human. It should be like Homo sapiens, but it will take into consideration and then it is going to search accordingly. So that much sophistication is made. Then one can take...
Prashant sir.
Yes sir.
I have a doubt. This is Dr. Mahesh here.
Yes sir.
I was the one who asked about the COVID viruses. So as I said, so there are limited sequences that are present in the databases, which is around 40. Yeah. And most of them are of similar length. So some 3 billion base pair also, the base pairs. And there is no variation amongst those sequences. And most of it is from Wuhan that I found. So I just wanted to see if there are any sequences that are present so from India or from any other state. I could not find much of the sequences. That's what I was trying to get the sequences of. Okay.
See, if again, the data comes to NCBI whenever a scientist deposits there.
Exactly.
So in most cases, people will publish making the comments like these mutations have happened or identified. Maybe at the expression level, from the expression they would have identified. But if the information is done at the sequencing, and if they store in the NCBI, then only we will get the, this is the problem with the public data. Even if you take any of the like vaccination data or anything, right? They will say like so many vaccines are done, vaccination is done, but we won't get the names or anything, even the protein structure. People develop the drug molecule, but the structure won't be available in PDB because they will not deposit. So now why they won't deposit is again a like funding issues or patenting
limitation, right? Structural limitation.
Why should I make this data public? Others will use. So what is the IP with me? So there are so many questions. So they will not deposit, but they will tell everything. This is how the disease is started, and this is where it moved to. So the whole thing we will but sequence. But what is that one can do is, once we have this complete genome information, right? We have this, let's say we have like 29,903 base pairs, single stranded RNA. And if in the paper, if they mention where the mutation is, we can manually change the nucleotide. We can manually induce that.
Okay.
That is the major way even we also work with. We may not get like tyrosine alanine mutation. That structure we may not get, but we will use the tool, change the amino acid manually, then use it for the study, docking or any of the aspects. So from the literature, that mutation information we will take and then manually make this mutation.
Fine, sir. Yeah. So anyways, I will discuss it in detail. So I would like to meet you and I would like to discuss it with you. So
yes, sir. Sure. Yeah. Any other questions or doubts? Okay. So if it is not there, now we will go for the other database. Okay. That is the, even in the morning also, professor mentioned about the Swiss prot, UniProt, Swiss prot. Now, when I typed here, human CCR5, right? If we recall with this, we got around 11,500 or 11,000, let's say 11,000 entries. Now in the Swiss prot is a universal, here uni refers to universal, prot refers to protein. Okay, that is why it is given like UniProt. Most of the databases names are kept in that way only. Okay, GenBank is a general bank, general nucleotide sequence database, like that. Now if I type here, maybe for you maybe coming out the visualization may be appropriate, but here it is not clear. But anyway, even in yesterday's presentation also, it is mentioned like Swiss prot and Tremble. Maybe tomorrow you can also explore it out. Why in the Swiss prot we have 5,65,000? Why in the Tremble? Okay. Why in the Tremble we have so many entries? Okay. So tomorrow again I am going to ask this. Maybe you can just tell me why there are so many less entries in this Swiss prot, and why there are so many entries with reference to Tremble. This is around 2 billion. This is around 5 million. This is 2 billion. Okay. So why there is so much of difference? There is a information already given there, but you can just explore it out and then tell me the difference. Now, if I type here human CCR5, how many entries I should be able to get? Any guess? Without doing, we got 11,000 here. When I type in the human CCR5 UniProt, how many entries I should be able to get? Any guess from anyone? Just some attempt. Now, UniProt SwissProt is a primary or secondary database? That we discussed.
So secondary database.
Secondary, sir.
Correct. Now, what is the secondary database refers to?
That means it is based on primary database that has been processed, that is curated like this.
Perfect. So the secondary is derived from the primary database. Now, when I type human CCR5, when they are derived it from the gen bank, which is 11,000 here, so the entries should be less here. How many it should be? Around some approximate? Some guess.
Some 200.
200. Anybody else? Now, the beauty of this UniProt, right? That's where even in the yesterday's presentation, I mentioned you got to appreciate the work done by others. Okay, so people have curated, annotated in such a way that they reduced this 11,062 into 1. Okay, it is as simple as, again, if I put it back into the terms, there are 11,000 students provided the information to the chairperson about the, let us say, the university. Okay, because in university we can have like 11,000 students, alumni and everything together, for example, because it's all related with one thing only, human CCR5, University of Madras. If we take, for example, easily 11,000 papers we can get. But now all these 11,000 entries were analyzed by set of people and created a one page which explains about this human CCR5. Okay, so there is only one entry related to human CCR5. Second one is mouse CCR5. Another one is chimpanzee, I think, or green monkey. Another one is another variety. So there is no duplication at all. Nowhere you will find the human CCR5 again. We may have like CXCR or CD4 or CCL4. This is different than this. Now people have curated in such a way that the entire information about this human CCR5 we can find it here. I don't know why it is not visible clearly. Let's use Chrome. I hope for you it is visible, right? In the blue color will be there in the left side. Okay, I'm going to place it here. Now this is how exactly it is going to look. Now you have a clear information. CCR5, it acts like a receptor for number of inflammatory diseases and also acts like a co-receptor, CD4 being the primary receptor for the human CCR5. It's very clear just in two such sentences. Now on what basis they said like it is in receptor for? We have a reference. What made them to say like acts like a co-receptor? We have 5 publications where they said like for each and every aspect functionality they are going to report here, there is a reference, there is a source or there is a reference for everything, and there is a hyperlink given. As in the morning it was mentioned with the KEGG, right, or the different databases mentioned. So it is not required to remember all the database names. If you just remember UniProt, that's more than enough. Then the systematic classification of the data is made, function related, taxonomy related, subcellular location related, where it is present. It is there on the outer cell membrane. It's clearly highlighted. Pathology related, we have all the involved in this, right? This is susceptibility, mutation and its effect. Everything is organism specific information, then post translational modifications, expression, interaction with other proteins, structural information, whether the 3D structure of this protein is available or not, family and domains, sequence. So how many amino acids are there? It is having 352 amino acids. Yes, the length is 352. Now, before you come for the next class tomorrow, okay, I just want you to do a small homework. When we did with this, right, we took this particular gene coding sequence, right? So we also know that one amino acid requires 3 nucleotides. Okay, if it is not known, you can also do this. For one amino acid, even in the yesterday's presentation, right, Devaraj sir mentioned genetic codon table, and then one amino acid with the duplicates codon table. So when we know that the coding region starts from 13 to 1071, you can also make a note in your notebook. 13 to 1071. How many base pairs it is coming out to be? Okay, from here to here, if we count, what is the count? How many base pairs it is coming out to be? If one amino acid requires 3 codon, for example, ATG codes for methionine, then GAT codes for D, aspartic acid, then TAT codes for tyrosine, like this. So how many comes out to be here? And if we divide this by 3, we should get 350, 352. Now, are we getting exactly, let's say for example, even if I want to make the process simple for you to do, 352, if I do 352 into 3 is 1056. So are we having 1056 base pairs or less or more in this? Okay, that you can check, and maybe you can let me know whether we have 1056 or not. Then along with that, okay, spend some time in looking into this mutation information, natural variants. So the entire information about this human CCR5 cross-reference to again GenBank. Okay, if I click this, it will go back to the GenBank. If I open here NCBI, so we will get back to the GenBank. Okay, so KEG, linkage, protein information. So what I want you to do is, maybe you can spend some time, okay, and then you can let me know what is that they are missing. Okay, is there anything that is not covered about the protein? Okay, is there anything which they are missing to cover about the protein? If we feel like more or less everything is covered, and for everything there is a reference, right? Publication information, hyperlinks. It takes you to the different database. Therein you can work and execute. Any doubts or any questions related to this human CCR5 or UniProt majorly? If there is anything, please make a note of that. Okay, maybe tomorrow we can discuss and address that.
Tomorrow is a holiday, sir. Tomorrow is a holiday. Day after.
Yes, day after tomorrow, sir. We can do it.
Sir, I have one doubt, sir.
Yes, sir.
Hello.
Yeah.
Sir, earlier you have showed we can search for this human CCR5 in NCBI nucleotide, right?
Yes, sir.
Instead of going there directly, we can use this UniProt section, right?
Exactly. True. So what we do is come to UniProt, okay, get the interaction or information, and then from here we can go to GenBank. Reverse engineering. We can do it in a reverse manner.
So that reverse going back to GenBank is for the purpose of getting this faster sequence only, right?
Exactly.
Yeah, here we don't get this faster sequence in
Yeah.
UniProt.
Here we will, because UniProt is a protein sequence, so
Okay,
we'll
okay,
get the pasta of the protein.
Okay, okay.
Yeah, from here we can take and then further use. But if you want the DNA sequence, so from you will use the cross references, and if you want EMBL or GenBank or DDBJ, the database we can select, and then we can go whether we want the genomic DNA or mRNA, what do we want. So accordingly we can select, then extract it.
Thank you.
Yes, sir. So if there are any doubts, as I mentioned, we will. Oh, now it was hiding behind. So we will take it up in the next class, sir. Okay. Okay, sir. We will stop here, and then we'll continue in the next class.
Anybody